Mau di extend apa duplikat?
Nggak tahu saya. Enaknya gimana?
Dari ini tempat ada
Iya.
full screen.
Enggak, ini saya kan dari Google Docs, Google Drive. Mending di Google Drive atau PPT?
Ini langsung tampilan
Harus di download dulu ya. Ini berarti
saya mandu untuk tampilannya. Iya.
nanti setting saya sih. Nanti ya asal saya atau tim saya lah.
Cek. Kemudian scattering ya. Klik tom ya.
Saya lewat komputer.
Tampilan sini langsung ke
All right. Looking good, everyone. All right, yeah, you guys, yeah. These seats are for the judges. This one is okay. How are you doing? Okay. All right. Who do we have here? D42, looking good. D30. Who's our D30 team? What's that? Oh, okay. We're waiting for that. Okay. Okay. So we're waiting to flip the coin, and that's how we'll know who will be first. So I just want to make sure that the projectionists, they understand at the beginning of the tournament, we're going to do a coin toss, and that will determine who goes first, right? Okay, and then they will, okay, excellent. And the judges, your software?
So use the computer or
Either way, you can use whatever you're more comfortable with. You can use Microsoft
yes.
or you can yeah, okay.
You want to use
Oh yes, yes. So you're not permitted to have an electronic device
yes.
during the tournament. I forgot to mention that. I mentioned that on the thing. This is an exciting match. I can already feel it here. Okay, how are we doing? Judges ready?
No, not yet.
Your apps okay? Let us know when you're ready with your apps. And we have D30 and D42. So the projectionist, he can see those two. He's got those two. Okay, and the timekeeper. Who's my timekeeper here? Okay, so you let me know whenever they are overtime, whenever they hit that, okay? And also, when one's asking a question, make sure they don't go more than a minute.
Tampilan YouTube-nya.
I'm the referee.
Oh, okay.
I'm the referee.
Sama
Right. Yes, I'll do, but I'll take this one. Okay, all right, great. So the first thing we're going to do, I'm going to flip the coin. So heads is, who's that on the 100 piece? Who's on the head? Who is that? It's some person. Okay. Anyway, so that's heads there. This is a real coin. Head or tail. I'm going to flip this. I'm going to say, you're going to make the call. Okay? So there it is. Okay. What's the call? Heads, and it is tails. Okay, you have the option. Do you choose to go present first, or present second? Second. Oh, strategic. Okay, so D42 is going to present first. D42 is going to present first. Okay, so now, hold on. Okay, he's connecting. Give us a second. The judges are getting there. So you're going to be presenting first.
Yeah.
You'll have 2 minutes to do the presentation, and then they will count you. Okay. So just keep focused about what you're going to do. If the question is not clear or something is you're going way over time, then I will intervene as well. And so just let us know, judges, whenever you are ready, and then we will start. Either way, when you're presenting, whatever you want to do, or even when you're challenging and you want to stand up, that's okay too. But you just can't leave the room. During the match. Okay. Good. You want to see? Okay. Hello, 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 hello. Are you working? Hello.
Hello, hello. Oh, okay. Okay. Hello, hello. Oh, okay, okay. Here we go. Okay. Okay, let me know when you guys are ready. Okay. You want to give a test? If you give a point and subtract a point, can you do that? Okay, great. Okay, great. Okay. Well, let's follow. Is it working if you award a point? Okay, great. Is that working if you owe it? Okay. Go ahead and test, yeah?
Oh iya, Pak. Tolong dibantu, Pak, untuk
Okay, great. You're able to add? Can you subtract those as well? Nice. Okay, so you guys are okay? Looking good? Yeah. All right. Timer, are you ready?
Okay.
Okay. Go ahead. Go ahead and start. Timer.
Good morning, everyone. We are from the 42 team, and our research is about unveiling anti-thyroid cancer potential of Augustemon Cabin and Muria Kongi, network pharmacology, structure-based virtual screening investigation, and RNA sequencing from Indonesia patients. Next. Actually, our research, it has two main idea. The first is we want to find which natural compound that can treat thyroid cancer, because thyroid cancer is still the cause of death in the world. And then thyroid cancer, it has two types, and in our research we focus study about PTC, papillary thyroid carcinoma. Why? Because PTC, it has a bigger cause of death compared to other type of thyroid cancer. And also in the thyroid cancer, it's still challenging to treat using several treatment sites like surgery and other treatment. And then the second, we want to find which mechanism that targeted by the natural compound. And then we want to know the signaling, especially in the RAF-MEK mediated pathways that is correlated also with mortalin, because in the thyroid cancer still unclear how mortalin can upregulate in the thyroid cancer, because thyroid cancer is high regulate in the thyroid cancer, but there is no evidence and no publication until now how mortalin could increase in the thyroid cancer. And then the next, in our study, we are using bioinformatic approach. So there are several steps in our study, like data collection using LCMS, GCMS,
15 seconds.
and then others.
How many seconds left?
Next, please.
Okay, 7.
Next, please. 242 genes that's related to PTC, and our results just like this.
Okay, the times is up.
Okay, thank you so much. Okay, over to D30 for your challenge.
Okay, thank you for the times. I have a question. Where do you use the why do you use the plants to obtain the ligands cleonasterols, norm, and ena and coenibin? Jika gimana? Kalau misalnya dia.
Have you ever tried to find those compounds in other plants? Because if these compounds found in other plants, maybe you can just use that plant. Not difficult to obtain from this.
Yep. Okay. Thank you. Cleonasterol and norn-12-in is the common compound that found in other plants, but coenimb is specific found only in Muraya koeknigi or temuru. Temuru or daun kari is a common herb recipe that used in Aceh. So we examined and developed this temurui or Muraya koeknigi as an alternative for PTC, because this one natural compound. Thank you.
The second question is, did you know the specific cancer stage have the different expression genes on the maybe the profiling proteins? Do you determine the specific target at the stage of the thyroid cancers?
Based on there is professor in the USA. Her name is Professor Jong-in Park. So he studied about this thyroid cancer, and I was involved in this study. And we have data, but we cannot show in here because it's unpublished. And the data is shown that mortalin is increased in all stage of the patient. Thyroid cancer patient, and then we have tried also to treat cancer using combination of natural and also synthetic compound, and actually how it can treat the patient is different of the stage. For example, in the stage 1, maybe mortalin could decrease using a specific dose, but if it's stage 4, it's maybe we need higher, and also maybe combination with other treatment. Yeah, thank you.
About the structure of the protein that you use as receptor, I concern about the accuracy of it and the accuracy of all of the software that you use in here. How about that? How you can because in silico is predictive, right? And how much it's accurately this result is.
Okay, thank you. Can you go to the next slide, please? Second. Okay, so we do the replica techniques for doing the dockings. There's 3 types of docking that we use to all of the compounds that we examined, and we found that the top 3, coenimb, cleonasterol, and also 12-in, is the best average score from all of the analysis that we do.
I'm not asking about the docking score, but the accuracy of the protein structure that you use, because I concern that the structure that you use is from modeling.
Yeah.
Is it right?
No, from the AKT one we use 3069 code from PDB-ID, and it's what's that name is X-ray diffractions model, so it's valid enough.
How much is the RMSD?
I'm not sure, but it's between 2 until 3 angstroms.
Because the RMSD should be below 2 angstrom, right? Yeah. So.
Yeah, I think AKT1 has some different model, and we can found it from so far. So we cannot decide. I think we cannot decide the most precise AKT models for all of the proteins in the human's body. So we confident enough that all of the analysis that we do using the some valid and precise data from the modelings beside its in silico approach.
Can you show the reference of the accuracy of the protein structure modeling and the software that you use in this research?
Yeah, I think for all of the model that we use in this data for the proteins model, we do not include this in these presentations. But can we go to the slide before for all of the tools that we use for the bioinformatics analysis? One more. One more. Okay, yeah, in here we use. Oh no, next one. Yeah, we use Autodoc Vina for the molecular docking and ligand preparations. All of the analysis using Vina in Ubuntu, and we found all the data are correctly related enough to the
50-second
natural, to the native ligands that as inhibitor as AKT1. Thank you.
Okay, I have a question. You know the natural compound like the I see in your elegance, please. Now, this is the natural compound that you know, but the natural compound is not synthetic from human synthetic, and your target protein is inner and in the inner of the cell, such as the IKT is
15 seconds left.
transcription factor. How you validate your target can reach the target, your compound can reach the targets inner the cell.
Yeah, the limitations of our research just in the in silico.
Okay, go ahead and answer.
Yeah, the limitations of our research just in silico analysis in the molecular docking, and the next step is molecular dynamic simulations. So I think it's not proper to ask some validations in the in vitro on in silico because our research just limited into the in silico approach. Yeah, and here our final research just some model that predict this compound from both plant Muraya Koeknegi and also Pogostemon cablin can be an alternative to the PTC cancer. Thank you.
Okay, thank you. I declare the end of the first round. Okay. You have 1 minute to review, decide what you want to do, what your strategy is. So 1 minute from now.
Time's up.
Masuk di situ yang sudah gabungan dari kami atau?
Iya, yang di sini 5, 6.
Jadi gabungan atau?
Itu udah total.
Total berdua.
Yes, yes.
Karena dari Victor itu 3 dan 4.
It's supposed to be the total of both of you.
Right. So I think it should be more than 6.
Okay.
Right. Because you have, for presentation, the total is 3. For each round, you can get up to 2 per team.
That's correct.
So 6 seems really low for a total.
Okay, hold on a second. We have like a technical.
Okay.
I'll be right back. Okay, compose yourself for it. That was excellent round, by the way. Timer, are you ready for our next? Let me know whenever you're ready. Okay, D30, are you ready? Okay, you may begin.
Okay, good morning everyone. So here is our topic about bioinformatic analysis of microRNA mediated regulation in their potential as therapeutic targets for lung cancer. Next, please. So based on global cancer data, lung cancer has become the most lethal cancer. Previous studies show that current cancer treatments such as chemotherapy and radiation still face high mortality rate. Therefore, we need an innovative treatment for lung cancer. Previous study has shown that microRNA can act as tumor suppressor. With the help of argonaute protein, the microRNA can degrade the mRNA. So from this background, this research aims to identify the most potential microRNA as the novel therapeutic approach for lung cancer in stage 1 until 4. This research utilized the microRNA data from TCGA database and analyzed it with bioinformatic approach. Next, please. So the heat map and ANOVA analysis show significantly different expression level of microRNA between the normal and cancer sample. Meanwhile, the volcano plot split the microRNA based on the expression profile. The table shows the list of the most potential tumor suppressor microRNA in stage 1 until 4 and its target gene. Next, please. And the selected microRNA have been evaluated with ROC analysis. Meanwhile, the target genes shown to be involved in the pathway and biological process of cancer. The molecular docking show highly binding affinity between the microRNA, mRNA duplex, and argonaute protein. This is the visualization of the docking. So the conclusion of our research is these 4 microRNA have potential to be developed as therapeutic treatment for lung cancer in stage 1 until 4. However, we still need to validate this research with the molecular dynamic and in vitro and in vivo research. Thank you.
Very nice. Okay, open for questioning. D42, yes.
Okay, can you go to the previous slide, please? Yep. Yeah, some microRNA has its log fold chains or log FT, and we can decide it. But for the up and down regulated, can you describe how you chose the threshold for which log FT that can be up regulated and down regulated?
Okay, for the log FT value, we have to do the compare between the cancer's population and the normal population, and we also analysis between the age states of the cases 1, 2, and 3, and 4, and we use the threshold 2 because the previous journal I seen the journal before he said with he said the threshold of 2 is very valid and very strong significance.
You use TCGA data. That means you use all the public data. Can you confidence enough that all of data are in the normalization size? I mean, you combine all of the populations and all of the research. I mean, can there is a valid confidence score that all can be normalized?
Okay, the TCGA database use the all the population of the global race, like the black people, white people, and Asian people, and the others race and ethnical. And the TCGA analysis use the we use the normalized like the scaling and scaling and normalization to normalize all the population of the sample.
Okay, thank you. I want to ask, is there any previous publication about mRNA, non-mRNA to target the lung cancer? Have you ever searched about it? And then how is the previous publication talking about it?
Yeah, there is a previous study about this, but it's not using this methodology. And yeah, the microRNA that they found is similar like what we found here, but not all.
What is the difference? I mean, why you want to do your research if there is previous research published about it? What is the novelty for your research?
Because we use the new methodology, we use bioinformatics to analyze all of the database in the CGA database, and the previous study have not studied about the TCGA database in there, and in this research we analyze in each stage which microRNA that is the most potential in each stage, and meanwhile, in the previous study, it doesn't research about the microRNA in each stage.
Okay. You said before that you study about this, that you want to your RNA will be the tumor suppressor, right? How about the previous study? Did they also talking about that, and then what target or what downstream? And then will you analyze also the downstream of your tumor suppressor? And then, yeah, what's next step?
Actually, because we still have the steps that we have not done in here, like molecular dynamic and plasmid design. Maybe we want to focus on that first. But also the wet lab like in vitro and in vivo is needed.
Okay.
And
okay, you said that you want to do knockdown or what?
Yes.
Plasmid design for what?
For knockdown.
For knockdown. So, what method that you want to use for the knockdown system?
Okay, maybe the next research we use the maybe the plasmid knockdown based on the CRISPR-Cas9. So
CRISPR-Cas9. Okay.
But actually, we not research too much about this CRISPR. Maybe next time I will improve this research.
Yeah, you say that you will do the knockdown CRISPR-Cas9 analysis. But in some data, not all the microRNA can be expressed. I mean, there is completely difference between populations, age, sex, or anything kind of race. Can you make sure that the CRISPR-Cas9 can be, just targeting the valid expressions of genes that based on your analysis?
Well, because in this research we only we want to know which is the most potential microRNA.
30 seconds.
For that question, maybe it can only be answered with the next stage of research. So yeah, but in this database we use almost all race and ethnicity. Yeah, so.
Yeah, but there's something interesting in all of the populations that use, because sometimes there is some significance factor in all populations. I mean, just like between male and female, there can be a lot of difference.
What's your question?
Yeah, my question is just like why you use all of the kind population, sex, and age, because there can be a significant difference between all of that factor.
Yeah, we acknowledge that we still need more research about that. How to differentiate between the, for example, female and male.
Okay.
Yeah.
Okay. And second, because this is lung cancer's patient, actually the balance between the female and male in the TCGA database. And this is decided used to all population of the sex.
Okay. That's time. Thank you so much. Okay, good job. Okay, judges, go ahead and submit your scores, and they will tabulate, and we'll tell you who won this match. But you guys did a great job. Great opening match. Very impressive. Very impressive. Nice work. Both of you, this is the first round, both of you are going to be moving on, but yeah, let's see, wait for the suspense. Thank you. You have it. Okay. Oh my gosh, what a close score. So the winner is D30 by a point. Congratulations. Congratulations. Okay, and congratulations to you. Very good match. Awesome match. Good job. Okay, so D30 actually, and so thank you so much. The next team is going to come. You can go in the other room and see who your next opponent is on both sides. Great job. I would suggest, because you see the scoring there, if you want to redo your score a bit, so the next time the title, if you can move the title a bit, so that, you know, I mean, we will try to adjust it. Just keep that in mind. And I would advise you also, both, just on your next one, I know it's difficult beginning, but just relax. Slow it down a little bit, right? Just focus on it, and but your content, your mastery bit, very good. The score shows it. So thank you. Good luck to you going forward. Okay. Next team. Okay. Who do we have up next here then? All right, D16 and D47. Okay, who do we have? D16, D47. Who's D16? D16? Yeah. D42. You're D16? Yeah. Okay, have a seat. We're ready for D16. Awesome. And opponent is D47. D47, are you here? Okay, come on up. Okay. That was an awesome match. That was wow, man. That was impressive. So you can actually take off, I guess. Yeah. Yeah. Okay. Yes, please, D47, please be ready. We are ready for you. Okay, do you understand the rules of the engagement? Any questions or clarifications?
Do we go first?
No, I'm just asking, any questions or clarification? Do you understand the rules?
Yeah.
Okay? Okay, great. Now, I'm going to flip the coin. I'm going to ask you to call it, heads or tails. Okay? Go ahead. Heads or tails?
Tails, maybe?
Tails, you choose tails. It is tails.
Oh, okay.
It is tails. Would you like to present first, or you want them to present first?
I think we can present first.
You're going to present first. Okay, they like to present first. So D47 will present first. Okay?
Is it the right presentation?
Yeah.
Okay.
Okay, hold on just a second. I just want to confirm, for the scoring, you guys are going to go on paper, right? So we're having a bit of an aptitude, yeah?
We will try to send.
Okay, okay.
Yeah, I think we know it.
Okay, great, okay. Excellent. So we, okay, are you ready on the timer?
Okay.
Okay, go ahead, start.
Let's start now.
Okay, good morning, everybody. So this is our topic, sequence homology-based enzyme detection of plastic degrading gene using PCR approach from enrichment of plastic contaminated soil in Galuga, Bogor, and Cipacang, Sotang. Next. So the background is because we know the potential to screen, find, and produce plastic degrading enzyme from new resources for an environmentally sustainable solution. And we want to search and prove the potential genes we have in Indonesia from our local environment. And because the founding of Pichenia bacteria saccharinaceae, or before we know it, Idoneola saccharinaceae, that can produce enzyme that work simultaneously between PTSA and MHTase. So we want to find if we have the same encoded gene. Next. So this is what we do. We went with sampling into different area, Galuga and Cipacang, and then from there we cultures our samples in minimum media with PET film on that. From the paletting, we extract the genome, and then we amplification the gene using this primer. After we successfully amplified, we sequence it and verification to data bank. And then the verified samples we send to 16S metagenomic sequencing to
30
see
seconds.
the compound. And then, so this is the result. This is the one that the gene that we successfully amplified. And the one from Galuga, we found MHTase gene for sequence, but from the Cao Tangyang, we found GPTase that we only found the partial. We successfully only the partial parts of the gene, but from the
Time's up.
From the 16S.
Okay, I'll give you 5 seconds.
Okay, from the 16S, both of the MHTase and PTase have the same classification from the state of the order that they came from Bucodiolas.
Okay, that's it. You're all done. Okay, thank you so much. Okay, you have 6 minutes to challenge, starting now.
Thank you. So I'm curious about the choice of choosing the landfill in 2 places. What is so special about these 2 landfills? Because you're looking for enzyme degrade plastics, right? Are there any proof that in these 2 landfills there are actually evidence of plastic degradation?
Okay, because our research only try to looking up because he come from south of Tangerang and I come from Bogor, so we just randomly choose that sites to choose the soil sample. Like in my city, Galuga is one of the biggest landfill in the Bogor and west of Java, I think. Yes.
Okay, so the question is, what is more important, the bacteria, the species itself, or the gene in that species?
Because mostly we are working with the recombinant enzyme, so we tend to have the gene instead of the microbes. And because for the genes MHTase itself, it's actually not the extracellular enzyme, so we have to choose the right host in the recombinant enzyme, which can produce it extracellular, so it more easily to get the enzyme rather than in natural microbes, because it's actually a lipopolysaccharide produced in the cell.
I'm wondering why not just get the bacterial pure from Japan and get it here. This probably even faster rather than finding our own Indonesian bacteria. Is that what you're trying to say? You have to find Indonesian bacteria?
Actually, in our lab, we already construct the same gene from the Idoneola saccharinaceae from Japan, but we also want to see, is it possible to find the same gene from the Indonesia? Just like random project, like we just want to know about that, the possibility of the gene spreading in Indonesia, because originally it comes from Japan, and we want to know if we just looking up in the soil sample like contaminated with plastic, can we possibility find the genes there?
Yeah, I guess it's just that the matter of finding the locations. Is there any evidence to show that in this certain landfills there are plastics being degraded that we can actually assess. Because if you can show this particular landfill has lots of plastic being degraded, it could be a lot of bacteria candidate actually living there, rather than just randomly taking a sample.
Because we went to the landfill that which contaminated a lot of
3 minutes.
different character of plastic like PET, HDPE, LDPE. So we want to know in the amplified the green that we try to extract, it is have the same capability that the one in Dionella saccharensis produce. I think we will have another different characterization of the enzyme because we come from a different region, different environmental condition. So the pH, I think the environmental will have a certain effect on the express gene in our area, and then it will in our perspective it will have more easier for us to maybe produce it on a scale up.
That's a minute.
Oh yeah. Yeah.
Stop. Okay. Go ahead. You have another challenge.
You said before that you choose a location based on your home-based location. If you have the opportunity to choose any location, methodically, what is the best methodology to choose a location, and what it will be in Indonesia?
Okay, actually, from the GB site, from the south of Tangerang, the landfill is really near with the recycling sites of the PET as of a plastic bottle specifically, and he just took the plastic from that site. So actually, the soil itself is really a bit specifically contaminated with the PET plastic, and in my sites. I also try to looking up for the
1 minute left.
PET plastic contaminated land that the land is specifically where the people working there put the PET plastic there. So I choose the land like in that site. But if you're asking us which site, actually the GB site is the good side one because it's the recycling place of the PET.
Okay, that's it. 1 minute. Okay, thank you. Next question. Yes.
But I guess from the conclusion, do you actually find the genes that you're looking for in that in your site?
Yeah, we successfully amplified the two genes here for the full sequence. But unfortunately, the PETAC gene that we amplified is only the partial one, so we don't have the full length. So we have to work on in that. So we have the full sequence that we can express
time's up.
later.
Okay, go ahead and finish your answer.
Yeah, we have to continue to find the full sequence of the PETAC one. Yeah.
Nice. Thank you so much. Very good round. Okay. So take a minute. Get grouped together, you will be presenting in a minute. So discuss your strategy, yes. Okay. Are you ready? So.
Okay, start now.
Start.
Bismillahirrahmanirrahim. So our work is the detection of DNA methylation from buccal swabs using NGS to study stunting. So next slide, please. So stunting is still a big problem in Indonesia, about up to 20%, but in Pandeglang, is a few hundred kilometers from Jakarta, is about 30%. And we know that chronic malnutrition is one of the major risk factors of stunting. One of the proposed mechanisms why stunting occurs is because when you have lack of nutrient, then the lack of methyl donor is important for reprogramming our DNA or the modification of our DNA using the path called epigenetics. So these epigenetics usually have this record in the baby's genome when the mother's having actually lack of nutrient. So we ask whether when the baby is born and they becoming growing up, whether the record of the epigenetics can be taken from a non-invasive specimen collecting called using a buccal swab. So and also to check this, we're going to use the NGS called nanopore to detect native DNA methylation, which is actually a good method to look at from small samples. Next. So this is actually our sample. We went to Pandeglang and we collect about 200 kids, 30% of them stunting. But we choose the highest 12 samples, 6 normal, 6 stunted, has a good quality of DNA ready for DNA sequencing using a nanopore adaptive sampling. So our outcome will be based on the stunting status, and we looked for the predictors that are the targeted loci of different methylated region using NGS nanopore, and then we're going to look whether actually a differences in the epigenetic profile in this genome. Next. So this is actually the current preliminary data. So we look at one of the loci, the interesting locus is the locus in H19 IGF2 gene, which is important for the growth hormone. So we have the difference, if you look at the red arrows, there's a difference of methylation between normal and stunted children.
Okay,
this will be a good
time's
preliminary
up.
data. Thank you.
Thank you so much. Okay, the floor is yours, B47.
Okay, you said that these genes has correlated to screening within the children is have the stunting or no, like so how can it possible that other gene it's actually contribute to the state that the children is after stunting? Why you only state these genes here?
Good question. So we collect the normal samples and stunted, the children already being stunted. And then we collect both DNA from both group, right? And then the way we looked at why we choose these genes, we actually look at the whole genome, the whole methylome. But we look at the previous published data, because the previous data, they use DNA from blood. And they look at these genes, H19, IGF2 locus, and also these other genes. So we thought, can we replicate their data using our method? Because they use blood DNA, we use oral swabs DNA. Because if it's turned out to be true, then we should see the same thing. But of course, you're right. It could be a different site, but we start with a candidate first. We start with what other people has published, and then we see some interesting differences. Although this is a limited number of samples, we're going to collect more samples.
Because you said that these genes based on your study from others, like how is the possibility to find the new one? Like it is really new. Like others people don't have to find the genes. How is the methods to find the gene?
Right. In our genome, there are about approximately 30,000 methylated sites. So it's going to create lots of bioinformatics and statistical analysis to really find the differences. And then we have to go back and look at the phenotypes, because stunting is not just all or none, right? There's actually a range of height. So we're going to also have this quantitative analysis of the numbers of methylated sites and also the difference in heights. Not only that, we also checking the same samples of these kids hopefully next year, because we follow up the growth of their heights. Now we can see whether the difference we see currently can actually project to the growth of these children. So in order to see whether there's a causal effect, we're going to follow up the children not only from this year's sample, but also for the next year's sample.
I'm curious about the sampling, because it's located in Pandeglang. That I know that West Java is have the highest stunting percentage, but do you have any like background or data why precisely in Pandeglang it has highest stunting rate?
Right. So one of the issue in stunting studies is also
3
socio-economic
minutes left.
factors. So we know there are socio-economic factors in these families that have problems compared to Jakarta, for instance. And also the educational level of the mothers could be also confounding factors. But apparently the numbers of stunting there is an intractable problem. I mean, the national survey is for 22%, but in Padang it's up to 30%. So this is actually very important information for the local health authority to really have to do something. But we also have this program with the intervention with nutrition. So the reason we choose this one because the highest numbers of sampling, so we can get lots of subject with stunting phenotype.
Okay, I would ask you about the limitation of your sampling, like how about the ages and why is the range ages is qualified for the finding this gene? It's accurate, and you said before that one year is not enough to find the genes. How to say that 2 years is enough or 3 years is enough? How to say that?
Thank you. We plan to do this study for 7 years. So for 7 years, because we along a prospective data, so we want to follow up all the way even to go to school, so we can also look at the cognitive data. So this is actually a baseline level, and we also have some preliminary data. Those kids were initially last year was actually normal, they became stunted this year, and those who kids were stunted last year they became normal this year. So it's a very complicated ones. So we are not pretending that we're going to solve everything, but at least we want to also prove whether the epigenetic program during gestation period, the first 1,000 days actually fixed, or you can actually change. Because the terms epigenetic is changing, right? But is it really changing as the kids are actually growing? So this method is very important because it's very precise. You can actually pinpoint which nucleotides actually being methylated from the same children.
Let's say after we know that these children have stunting status, like how to overcome that status, because as I know before that stunting can be overcome during the pregnancy or the after birth.
Yeah, that's true. So one of the things why we are interested in looking for the stunting on the epigenetic of the stunting children is to look at the possibility of some imprinted genes that are important for metabolism, because many of the stunted children, they are very high risk of obesity and also other metabolic disease as they're growing up. So if we can, because we also collecting their
20 seconds.
their weight, you know, if they're obese or not. So then we can also predict if they're because they're already stunted, right? But whether they're at risk of any other health problems later in their adult life.
One more challenge?
Oh, okay. Can I?
Yes.
Okay.
Time's up.
Sorry. Time's up. All right. Good job, teams. Nice work. Give us a few moments to verify your score, and we will let you know the winner. Nice job. Give us a few moments. We're just doing the verification to certify your scores.
It's like reading half a
Oh, jadi segini juga.
Nanti
ibunya betul makan nggak sih ketika dikasih
Iya, iya.
Nah,
Soalnya
itu kita cek dari anaknya berarti karena kan bisa aja dia makan tapi nggak diambil.
emang mesti long time ya Pak, maksudnya kita mesti
akan tracing juga.
jadi mulai dari misalkan dari ibu dicek, nanti anak misalkan keturunan pertama, nanti ibu seiring pertumbuhannya dia gimana.
Ini pernah nyampe ngecek ibunya juga.
Nah, kalau ibunya kita masih belum kepikir, kita yang mau cek itu. Karena kalau ketika dia makan, itu kan
Hold on, almost there.
ibunya nggak terdampak. Tapi mungkin kita bisa cek sebetulnya nanti kaitannya dari motivasi ini sama infeksian.
Karena kan setahu saya pas saya belum dulu, maksudnya kalau epigenetik itu kan dia berubah seiring perkembangannya si anak lagi. Nah itu maksudnya saya
kita betul nggak sih belum cek lagi.
Boleh di luar Pak? Enggak. Seru banget ya.
Okay, so these are your final scores. So we declare the winner of this round is D16. So congratulations. Congratulations. Thank you so much. Thank you so much. Okay, and you can go in the other room and you can see where you've advanced. So good job, and make your modifications that are necessary for the next round. Okay, now we're ready for the next group.
So they do not know the score yet?
That's the score.
Okay.
That's the score. Right there, yeah. 28-17. Great. And so our next two. D12 and D30.
Yes.
Okay. Okay, here we are. Okay, D12 and D30. D12, D30, D12 and D30. Ah, D30. D, D30. Is that you? No. D12. Okay, Trina, okay, come on up. D12 and D30. D30.
D14 ya, hitung sebentar.
It's confusing.
You're next, okay.
Yeah, this is day 12 and 30.
Right. Yeah, hold on a second. Maybe,
Di sebelah sini. Ya.
after this. So, okay, please take a seat. D12, take a seat. D12, take a seat. Okay. D30. Okay, take a seat. They're not here? Okay.
One hot, one cold. Second round is balanced. Yeah, soalnya kan ini
Okay, so all the teams, as soon as your round is over, check the brackets so you know what's the next round that you're in. So, confirming, okay, you guys are up again. This is round 2. This is your first one. Okay, so. Okay, are you ready, timer? Okay. Okay, so the first thing we're going to do is we're going to decide who actually goes first. Judges, let me know when you're ready. Okay, so I'm going to flip the coin, and you call it if it's heads or tails. Here I have a coin, heads on one side, tails on the other, okay? And you call it, go ahead. Heads, and it is tails. It is tails. Okay, so you decide. You have won the toss. You want to present first, or you want them to present first?
Second.
You want to, okay, so they would like you to present first. Okay. So D12 will present first. Okay. And, okay, let me know when you are ready, timer. Judges, are you ready? Yeah. This should be this should be updated for who the teams are. It says D12 and D30. I'll hold on there for the. Yeah. There it is. Okay. 12 and 30 is there. Okay, you're set, judges. Okay, you're set, timer. Yeah. Okay. All right. Start. Go ahead.
Good morning, everyone. We'd like to present our study. The title is Molecular Detection of Mycoplasma pneumoniae using syndromic testing respiratory panel in North Sumatra. Next. In November 2023, the WHO released a statement on reported clusters of respiratory illness in children in northern China, where eventually 75% of the patients were infected with Mycoplasma pneumoniae. In response to this, the Ministry of Health of Indonesia released an official letter to increase a nationwide awareness and detection capabilities across the country. So a cross-sectional study was conducted at our hospital between November 2023 to February 2024. Nasopharyngeal swabs from individuals suspected of respiratory illness in North Sumatra were tested using syndromic testing respiratory panel based on a multiplex real-time PCR that targets 22 respiratory pathogens. Next. So we collected 165 samples, 52% were male and 86% were positive. And then we can see that the most prevalent virus were adenovirus, and Mycoplasma pneumoniae was found to be the most common bacteria to cause the respiratory infections. In figure 2, we can see a very interesting findings that we found between November and February, within 4 months.
30 seconds.
We saw that the proportion of Mycoplasma pneumoniae were gradually increased from 2.2% to 7.8% to 11.1% and to 21.1%. So this study finding highlighted the importance of, so this study finding found that the increased proportion of Mycoplasma pneumoniae that has importance
Time's
in.
up.
Okay.
Okay. Okay, thank you so much. D30, the floor is yours.
Okay, so thank you for the presentation. So I see that in the title you use North Sumatra for the area, but in the sample you just use from one database from one hospital. So how can you assure the representation of your data with the title of North Sumatra? Thank you.
Thank you. So as we can see in table 1, most of the patients was from referral hospital. Why? Because most of the healthcare facilities cannot detect the Mycoplasma pneumoniae, and we were lucky that we have this tools to clinically diagnose, laboratory diagnose directly from clinical specimens. And then we accept from all cities that refer to our hospital. Thank you.
Can you assure the distribution of your sample can represent all of the North Sumatra, or how can you assure that?
So we basically do a consecutive sampling. So it's a total sampling from 1st of November and 27 February. So we just finished the collection data 3 days ago and analyzed the data, and we found this finding. And we hope to, that this data represents all patients, because we took everything that came to our hospital.
Okay, I have the question.
Yes.
Do you use the normal sample to compare with the Mycoplasma sample?
Sorry, say it again. Normal samples
Normal
of what?
sample, maybe in the normal population.
Do you use the normal sample to compare between the Mycoplasma pneumonia and the normal sample? The significance between it?
Oh, so we detected positive or so from all clinically patients with respiratory infection that we suspected have respiratory illness, we detected positive or negative, detected or non-detected DNA of Mycoplasma pneumoniae, and all positive results we included to our analysis.
How much is the prevalence of Mycoplasma pneumoniae with all of the sample?
So you can see here that the proportion is gradually increased from 2.2% and then 7.8% to 11.1% and then doubled to 21.1%. It's the proportion of Mycoplasma pneumoniae versus all other respiratory pathogens. So all other 21 other respiratory pathogens detected from the panel.
Do you have the biomarker to detect the pneumonia? To use the biomarker also?
No, this is a multiplex real-time PCR test. So it basically contains of 22 primer sets and probes.
3 minutes.
That can detect all of the 22 pathogens that you can see here, and all of them were viruses and fastidious that we normally did not detect it in routine laboratory setting. So this is very important because this Mycoplasma pneumoniae can cause outbreak, and it's often overlooked in routine laboratory settings.
Yeah, but I concerned about the significance of that findings because the count of the Mycoplasma pneumoniae is just 12, and it's just 1,434 per month compared to the other disease. So how is the significance of your findings, and how is the analysis that you use in this research? The statistical analysis.
So good question. So we just finished the data collection 3 days ago, and we found these results, and we plan to do an ANOVA to see the significance of the increasing proportion from each month, and which month is the one that is statistically significant by doing post hoc analysis.
Okay,
Yeah.
maybe I can suggest for your research to use the ROC analysis, because ROC analysis can compare between the normal sample and maybe in the disease sample, and you can have the sensitivity value and what is the efficacy of your biomarker can detect the specific in the pneumonia maybe.
Okay, thank you so much for your advice. We will consider it. Thank you.
No, but what's the defense? What's the challenge? What's he mentioning? Want to repeat that? Like, what's the specific challenge that you issued?
He recommend to use ROC analysis to
differentiate between the biomarker in the pneumonia. Okay.
Actually, what we plan to do is to take all the positive Mycoplasma pneumoniae and do whole genome sequencing. And so we think that distribution analysis using SPSS or ANOVA analysis to see is this proportion, increasing proportion from 2.2% to 21% within 4 months are statistically significant, and whether it's yes or no, we plan to do whole genome sequencing to see the macrolide resistance for this, because AMR genes, is an issue for this Mycoplasma pneumoniae. So that's what we plan. But thank you. We'll try to see if ROC suitable for this. Thank you.
Okay, time's up. Thank you so much. Okay, so that's end of the first round. Okay, good job. Okay, we have a minute to strategize. Yes. Bring up their Okay, D30. The floor is yours. Start. Yeah.
Okay, good morning everyone. So here's our topic, bioinformatic analysis of microRNA mediated regulation in their potential as therapeutic targets for lung cancer. Next, please. So from global cancer data, it is known that lung cancer is the most fatal cancer. Previous studies show that current cancer treatments such as chemotherapy and radiation still face high mortality rate. Therefore, we need innovative treatment for lung cancer. Previous study has shown that microRNA can act as tumor suppressor with the help of Argonaute protein. The microRNA can degrade the mRNA. So, this research aims to identify the most potential microRNA as the novel therapeutic approach for lung cancer in stage 1 until 4. This research utilized the microRNA data from TCGA database and analyzed it with bioinformatic approach. Next. So here's the heat map and ANOVA analysis, which show the significantly different expression level of microRNA between the normal and cancer sample. Meanwhile, the volcano plot split the microRNA based on the expression level. The table shows the list of the most potential tumor suppressor microRNAs in stage 1 until 4 and its target gene. Next, please. The selected microRNA have been evaluated with RLC analysis, while the target gene is shown to be involved in the pathway and biological process of cancer. The molecular docking show highly binding affinity between the microRNA mRNA duplex and Argonaute protein, and this is the visualization of the docking. So the conclusion is these 4 microRNAs have potential to be developed as therapeutic treatment for lung cancer in stage 1 until 4. I think that's all from us. Thank you.
You have 10 seconds if you want to say anything else. Okay, that's the second is over. Okay, thank you. Okay, fantastic. Okay, over to you, D12. You have 6 minutes to challenge.
Okay. Thank you. Very interesting research result. If you want to develop the microRNA to treat the lung cancer by increasing the expression of the expression of the miRNA to degrade the oncogene mRNA, you have to do the preclinical trial. So what do you think is the suitable animal model to do this, to develop this research? Because we need to see the side effect of this use of miRNAs.
Okay, microRNA have the lot of the target, not the one target in microRNA. The microRNA is target the maybe 100 genes and maybe in the 1,000 genes. This is the difference between the type of the microRNA. And then we not use the in vivo belum. I have not used the in vivo because it's already the in silico to have the potential, and maybe next time we use the in vitro and then in vivo to maybe validate the bioactivity maybe and the others side effect more the microRNA, because the microRNA is have the lot of the targets in the cells.
Yeah, so actually we just conduct this research in a week ago. So it is just the initial research of us, and we need a lot of steps for this development of microRNA.
Okay, maybe next week.
That's a minute to respond to that. You had a minute to respond to that. Yes, go on to your next.
Okay, a very interesting study. My question is, how will you do the bench to bed of the application of your finding to the patients? And second question, can I second or?
You have another 45 seconds to
Okay. A second question is, how will you be sure this Mirna, as you just said that the Mirna has a lot of cell targets? How will you be sure that your Mirna, which is potential as a treatment, therapeutic treatment, wouldn't be an off target? Thank you.
Okay, we can go to the previous slide. Okay, yes. This is we analysis with the total text context plus scores. This is the bioinformatic and statistic value that can make sure between the microRNA and the your targets. Maybe we can example FEGF in endothelial growth factor alpha. This has the best target of the Mirna HSI humans Homo sapien Mir 383.
3 minutes.
Total context plus score evaluate what is the type of the microRNA, because microRNA have the commonly have the 3 types: egg mer, 7 mer egg, and others. And this calculation between the type, maybe, and second is the complementary and the total GC.
Okay. Thank you. Do I still have question? Okay. Do you think, so currently lung cancer was treated with radiation, surgery, and chemotherapy. So what do you think, if you apply this alternative, will the clinicians or the patient will still needs the current treatment options?
Yes, because, can you go to the previous slide? Yeah, so this is the reference from us. We have a study that the chemotherapy and radiation still face high mortality rate, specifically in lung cancer. And this is the reference, but maybe we cannot open it in here, but you can read the reference paper. So yeah, because the current cancer treatment still face high mortality rate, we need an innovative treatment, and one of the alternative is this microRNA as tumor suppressor.
Yeah. If you use the microRNA in silico, and then you will continue to the preclinical trial in vitro, how will you overcome the human immune system if you use this potential mRNA to in human or in vivo?
Yeah, actually, we cannot answer it yet because it is just the initial. But we have our previous study. Can you go to the next slide? Next slide again. Next slide again. 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 It's a bit again. Yeah. This. So the previous study, we have researched the previous study that also show that the microRNA that we selected in this research has been researched also in other study that it act as tumor suppressor. You can go to the next slide again. So this is the stage 1, this is the stage 2, and again.
You have 2 more seconds to answer this question.
Yeah, this is the stage 3 and stage 4.
Okay, time's up.
That's it? Okay, thank you so much. Good job. Very nice. Round 1 and round 2. We will be going through and validating your scores and posting it. Give us a few minutes and we'll announce the winner. Okay. Thank you so much. Have a seat until the winner's announced. Have a seat until the winner's announced. Have a seat until the winner's announced. Thank you so much. Yes. Thank you. You can use the time to sort of decide how you can improve. Okay, but that was a good round. Yeah. That was very clever. You had those slides ready.
Okay, we have the total scores.
Okay, so here are the scores here. So the winner of this round is D30. So congratulations to you. Don't forget her. Was this your second match or this is your first match? Okay, so continue. So congratulations to both of you. Congratulations to D30. Nice match.
Thank you.
Okay, and go check who you're up against next and be ready for that. Okay, next we have who?
We have 16 and 14.
16 and 40. D16 and D40. Okay.
Yes.
D14.
Yes.
Are they? That's you guys. D14 and wait, is it D16 and D14?
Yes.
Is that right? Okay, D16. D16. Team D16. Team D16. Okay, so don't forget, no handphones or computers allowed to be with you during your competition. Okay, any questions on the rules? Okay, so I'm going to flip a coin to decide who's going to make the choice. You can see there, that's heads and tails. I'm going to flip this, make the call. You will make the call, okay? Heads or? Heads. And it is heads, as you can see. It is heads, in fact. So you decide, you want to present first or you want them to present first?
I think I would like to present.
You want to present first. Okay, great. So D14 will present first. Oh, sorry. Go ahead.
Soalnya berantakan kalau. Oke. Bisa kan? Bisa. Oh, sebentar ya.
And let's set up the scoring, make sure we have the scoring correct. So it's D14 and D12. Oh, sorry. D14 and D16, yes, thank you.
I think we can do this.
Next. How
Because it said that you can send it. Is that okay? Like this?
Okay.
Nanti next aja. You said it. Yeah.
Okay. Ya, saya setting.
Oh.
This is your first match?
Yes.
Okay. Good. That's their second one.
Yes.
It's all right. Just keep composed, both of you, okay? Just get ready, think of your strategy. Okay. Okay. So here we are, D14 and D16. Okay, judges, are you ready? Okay. Teams, are you ready?
Okay. Bisa. Ya.
So you are presenting, this is a PDF file, correct?
Yeah.
So then you will have to scroll to the next one, right? Okay.
Yeah.
Okay, so please, the time is yours. Start.
Okay, good morning. So our study is about Plantago ovata. So Plantago ovata seed husk contains mucilage polysaccharides, which has significant economic value. Unfortunately, the seed husk is only 25% of the total seed weight. So our aim is to increase the seed husk percentage by investigating the genes that are involved in mucilage biosynthesis in Plantago. Next. So what we did is that we compared the transcriptome profile of two genotypes, the wild type and the mutant Raya or Ray. Now this mutant has higher mucilage content than the wild type, and we extracted our total RNA from two different tissues, two different types of developing seed tissues, which is integument, INT, and then mucilage secretory cells, or MSC. We took our samples at two different developmental time points, 2 DPA and 4 DPA. DPA is days post-enthesis. And as for the method, we use RNA sequencing analysis. And our hypothesis is that the transcriptome profile in both tissues would be different between the mutant and the wild type at the same developmental time point. Next. And as for the result, we got around 25,000 genes expressed in our samples. And after further analysis, we found 18 genes in integument and 8 genes in MSC were expressed higher in the mutant than in the wild type. And those genes are involved in xylem biosynthesis, which is the main component of the mucilage. So we were paying attention. We were paying attention to those genes by doing the t-test. Next. And this is the result of the t-test. And from the t-test, we got 2 genes that were expressed significantly higher in the mutant. Thank you.
Time's up. Okay, thank you. Okay, the floor is yours. Team D16 for challenge.
Could you explain how the mutant was created?
Thank you very much. I think it's not a challenge. The mutant is already created in the first project, so it's by gamma irradiated mutant. Gamma irradiation, yeah, irradiated.
The reason I ask is because if it's actually affecting the DNA, why not looking for the difference in the genomic between the wild type and the mutant, and then look for what kind of genes being affected when they're mutated?
Okay, so there are 2 methods. We can use genome-wide, and then we can use RNA. But what happens is if there are changes in the DNA, it's not always changes in the RNA, the expression. So we are looking the expression first, and then after that we pinpoint which genes is actually upregulated, and then we can see after that doing the variation between these 2 genomes, because using the whole genome sequencing is more expensive than doing this RNA-seq data. Yeah.
Okay. So far, are there any, because when you look at the data, if you scroll back, because I don't see the expression itself is different between wild type and, excuse me if I'm not mistaken. Yeah, scroll back. Yeah, because the way you arrange is between wild type and, wild type and mutant, right? But in the boxes, it basically belongs to both wild type and mutant. I don't see a difference between wild type itself and mutant itself. Am I missing something?
Okay. You can see in here, there is directly comparison between wild type and mutant, because
Stand up and point if you want.
okay, this one, wild type mutant. The difference we can see in here, actually there are 3 spots, 1, 2, 3, but this one at the beginning, at wild type and mutant at the beginning, they are not really different in terms of the expression, but we already did the correlation, and then there is no really difference in here. I mean comparison, and then in here you can see this a little bit downregulated and upregulated, and in here this is a really strong signal, and this is upregulated as well, but it's not really comparing to this by signal. And then we do the t-test.
Okay, looks like so the integument and then the mucilage, there are 2, I guess, different developmental stage, right? Yeah. So if you look at the ballpark, I think they're kind of similar between wild type and mutant, right? Even in 2 different, because they're in the integument phase and also the secretory cells.
Which one? This one?
Oh, okay, sorry. No, I'm just looking at my eyes probably not right. So this actually the two different group, right? Integument and mucilage, right? So this actually the wild type and the mutant. So if you look at from at this side, or compare this one and this one, both wild type and mutant overall they are very similar. Is that right?
So we don't here we don't directly comparing with the integument and the mucilage. That's the reason why we separate in here. So looking directly wild type and mutant, wild type and mutant, and wild type and mutant, wild type and mutant.
Right.
So we found this together because we do the several combination analysis. Which one that we can show really different in comparison, because at the beginning we do integument first, and then we do the time point, and then we do the genotype and the wild type. I mean genotype and mutant and wild type.
Okay. All right.
Because we want to find what gene is different between the mutant and the wild type.
2 minutes.
Okay.
Yeah, not comparing the tissues, but more like comparing between mutant and wild type. What genes are different?
Okay. So it looks like this was interesting. I suppose this mucilage secretory cells are the cells actually producing the mucin, right?
Yes.
Yes.
If you look at the wild type and the mutant, definitely this is much higher here.
Yeah.
So actually, is it really significant? Your calculation is significant?
One gene significant. You can see it from the last slide.
All right. Okay. So now the question would be, how are we going to prove that this correlation actually is causing the increase of the mucin?
So based on the we are looking the xylose transferase biosynthesis. I mean, it's a produce that induce the xylem, and then xylem is actually the main component of the mucilage. And in another model species, there's affecting this, it can lead to the high or low the mucilage production. So based on the knockout gene in other species, the model species Arabidopsis. Yeah.
So this actually use the mutants created by X-ray, right?
Yes.
So if I just make a specific mutation in this particular gene, do you think would be sufficient?
I think for the two genes that may and I think it can lead to the higher production, because in the Arabidopsis they said that the mutation in IRX, upregulated in IRX can produce more mucilage. They specifically target the IRX14, but previous study they said that IRX7 it didn't we can't see in the Plantago. But actually from this project we can say that we found that gene expressed because we use specific tissue type. Because I think using the whole
Okay,
capsule
time's up.
will be reduced.
For this round. Okay, thank you so much. Nice work. Okay, you have a minute to strategize for the next round. Okay, judges, are you ready? Timer, are you ready? Yes. Okay, team. All right. Go ahead. Time is yours. Start.
Yeah, Bismillahirrahmanirrahim. So allow us to present our work, detection of DNA methylation from buccal swabs using next-gen sequencing to study stunting. Next. So stunting has been a problem in Indonesia, about 21%, except in Pandeglang, reached about 30%. And we know chronic malnutrition is a major risk of stunting. And one of the proposed mechanisms when you lack of nutrient is actually lack of metal donor. And we know sufficient metal donor is required for healthy epigenetic programming. So the DNA should get some methylation on the right side so it can express the right genes so it can develop normally. So our aim is to look at whether we can see epigenetic marker that's associated with the stunting using buccal DNA, because most of the studies in stunting, they use blood DNA from children, and blood is very invasive, while buccal is actually very easy to get. And the method we use is using NGS nanopore, because nanopore can detect methylation in its native form. We don't need to convert with chemical conversion, so we can look at the native DNA methylation. So next slide. So initially, the population has about 230 children. 30% of them are stunted. But out of these 231, we're going to look at the highest DNA with the good best concentration, and we select these 12, and 6 are normal and 6 stunted children. So looking at the outcome, we look at stunting or not stunted, and we look at the different targeted loci using this NGS, and we find an interesting result. Next. Okay, so we look at the several loci that has been shown in previous literatures. For instance, the H19 IGF2 locus, which actually encode the growth hormone, insulin growth factor 2. And if you look at the normal and the stunting, there seems to be a difference, because, for example, the normal has, if you look at the red arrows, then the normal has some signals, the green signals, whereas a lack of green signals in the stunted children. And we also normalize whether the error has been good coverage, and it seems to be this pattern is repeat with other genes.
That's 2 minutes.
Oh, okay, thank you.
Okay, thank you. Okay, the floor is yours for 6 minutes for challenge.
Thank you very much. I think your project, since the beginning of your project, the selection of the DNA is not correct. Why? Because this is based on concentration. I think you need to put effort to get the DNA, because this is only technical, in my opinion. So you have a problem in it's a small sample size. Why you choose only 6? It's not biological. There is no biological reason in there.
Right. So the reason we choose 6, because we choose 12, the best top DNA, because this is actually the first time in the world that people used we used to look at the DNA in the non-invasive in the buccal DNA. So if you look at the previous experience when using the DNA from blood, obviously there's lots of DNA. But when it comes to buccal DNA, nobody has done it, especially in children. So if we just put any DNA, then it's going to waste so many resources, and we cannot actually make any conclusion, right? So that's why we choose the right amount of DNA, at least similar to what previous DNA has been used. And in a way, we are surprised we can actually see the data. Initially, we kind of pessimistic, we can see a difference. But the fact that we can see difference is really interesting. Of course, 6 and 6, it's not that enough. But hopefully we can actually adding more, because now we know the optimization protocol using this method.
So your result is based on the 6 samples. Then what if you look at if we look at your poster, there is demography between age and sex. I have a concern because you use a different amount of number of the sex, and one of your genes, like IGF2, is actually a paternally epigenetically controlled. So I'm afraid that your result is not true, because between male and female there's a different pattern, and you mix, and then you don't have equal number between normal and stunting, and your stunting as well, the degree is different. Yeah.
Yes, that's actually a valid concern, and at the moment we are analyzing the data, because for the most important first actually is getting the technical data first, because if the technical data is not clear, because it takes lots of work how to set up the computational issues and also the analysis. We attempt to actually, the next stage is to look at the stratified according to sex and also to age, whether the pattern is stunting is actually hold true. But obviously, we're not going to stop here. We're going to look at with different genes, because we look at the different genes like this. Is actually this gene has been shown previous data, and we see a similar pattern. So in a way, it's a kind of that we are not really off track, because if it's really off track, then we can see any difference in this particular genes. But obviously, we're going to have maybe another independent ways to look at the methylation method to make sure that what we've seen here is actually real. But definitely, it's not going to stop for the biological analysis. We definitely need more samples.
So I can see here your decision. I mean, your conclusion is based on the visualization in IGV. But can you? I mean, how we can show that this is significantly different between a normal and stunting if there is no statistic comparison?
Yeah, definitely we're going to look at the statistical analysis later, because for the non-bioinformatician like me, visualization is definitely very easy to look. And that's actually the first thing we need to look at the data. If the visualization no difference, then there's no point of looking at deeper. But now we can actually pinpoint, we're going to calculate, because each bar actually representing each nucleotide, we can actually calculate, it counts how many differences, the percentage of methylation in each particular locus. So this actually is a start, but definitely we're going to need a very complex statistical analysis, because we have about 30,000 places in a genome that possibly methylated. So we're going to look, this is only a start.
Your choice of the genes, if you can see here, is 1, 2, 3, 4, 5. I don't see any comparison. I mean, you look at the whole thing overlapping in there, but how about if they are not different each other if we look at only one by one?
Yes, well, the bioinformation told me that there's some differences, but she hasn't actually sent me the actual data, so that's why I only show the H19 IGF2. But the reason I told them to look at these ones, because according to the previous literatures, and she already looked, there's some difference, but yet, again, for the full manuscript, we're going to describe the statistical analysis. But again, this is very descriptive in the very beginning, because the main project right now is to get around the technical issues, how to do this using this nanopore, because you can look at that native methylation. It's incredible, because you don't need actually to do the
30 seconds.
So if you look at the IGV, because you have several spots there, have you looked at whether or not the spot is really methylated, or what about the other comparison in the other paper?
Yeah. So these spots actually are the same spot where the location of the locus of H19 IGF2, which is known to be imprinted.
Okay, time's up.
Okay.
Go ahead.
Oh, okay. Yeah. Yeah, it's known to be imprinted between in males and female. So we're going to do this because it's a long read, so we can definitely characterize whether this imprinting, according to the previous literature, is also similar mechanism.
Okay, good. Thank you so much. That's the end of round 2. Give us a few moments to compile the scores, and we will announce the winner. Remain seated, please. Okay. That was a good match. Very nice match.
Thank you.
Okay, so this is the certified total here. So the winner of this match is Dean 14. Congratulations, very close match. Beautiful work on both sides. So congratulations to you, congratulations to you. All right. Nice work. Okay, so now there is a 10-minute intermission. and then we'll resume matches. So please check for the next your next match. Thank you so much. Nice work, everyone. That was very. That last one was very. Yeah, I agree with the so now we're going to take a break around a 10-minute break or so, and then the next round, get up and walk around. Yes, I would agree. Yes, it's getting better.
Nanti yang berbicara atau yang di briefing mic-nya itu paling jauh dihindari, karena tadi ada yang juga agak terlalu jauh jadi jomplang suaranya. Tetap masuk audionya, cuma kurang kencang ya. Jadi pastikan dia bicara, kan kalau dari sini enak kasih taunya gitu ya. Kopi dulu. Siap. Sama ini Pak, kalau ada yang bicara agak di close up aja. Oh iya. Chargeran mic ruangan di mana disimpannya? Ini lagi break sebentar, Ini dua laptop ya jadi.
Rong empat iki.
Lah kono.
Hah?
Iki paling berikut.
Lah pundi?
Hah?
Sebelah mana?
Entah ya, agak darurat.
Istirahat.
Oke. Tapi sama istirahat.
Tapi streamingnya di jalan ya? Tampilannya sih
Konfirmasi sama ya.
di kandung. Wong ngomong sik dhewe ana kan pintere nang wong jatuhan aku mau jalan di luar.
Manglet engkas.
Hah?
Manglet engkas.
Ya wis padha wae. Sampai jam 1. Jadi sejak iya lah, tuh, waktu nih. Wis waktu nih. Sarung ono ora apa iki? Ya lah nek. Kerja. Ada ini guli. Gue internet aku dia gue web, itu kok ini tapi gue web. Orang model web, iya, itu udah web iya.
Pelan kan?
Enggak ada yang mau web ini, iya. Website di copy ini kan nambah.
Dia lagi break, tapi udah mau mulai. Udah mau mulai lagi cuma 10 menit doang. Bukan break makan.
Coming up here soon.
Gimana mas?
Kita lihat jam.
Ready? C41. All right. Welcome. Fantastic. Yeah. Okay.
Cek one. Kalau mic ini masuk nggak sih? Cek masuk.
Okay, I'll give you a few minutes, yeah.
Oh iya ya. Oke.
Go ahead, yes, please. We'll wait for you. Go ahead.
Maksimal 5 sen. Maksimal ya? Iya. Kayak perlu pakai ini nggak ya? Pakai DJ.
Cek. Permisi. Ini.
Tes.
Nanti agak cek.
Pakai gini juga.
Nggak usah.
Ngerem. Mas kita menghilangkan ketegangannya.
Okay. How are we doing? You guys set? All right. You got your microphone there. You got your microphone there. Okay. Do you have any questions about the rules? Any comments, questions about the rules? You're clear on both things? We'll do 2 rounds? Okay. So I'm going to flip a coin. You can see this is tails. This is heads.
Yeah.
Heads there. That's tails. I'm going to flip this. I want you to make the call. Okay, ready? Okay. What is it? Heads or tails? You say tails. It is heads.
Oh,
See there, it is heads. So it is your call. Do you want to go first or second? Okay, so you will present first. So team D41 will present first.
Yes. Okay.
Hold on a second. We're waiting for the
Oh,
judges.
okay.
I said that. When the judge is ready.
Yes.
Hold on. Okay, but you will be going first.
Oh, my Juan. Okay. We're going to go ahead. He's going to be the referee, and I will, fill in as a judge. Oh, okay.
Okay.
Okay, everyone ready?
Go.
Yeah,
Go. Okay.
okay.
So you have 2 minutes. Yeah.
Okay, start now.
Good afternoon. We are presenting our project entitled Sputum Microbiota Profiles in Hospitalized Adult with Community-Acquired Pneumonia. Next. Imbalanced bacterial composition or dysbiosis in the respiratory system has a play role in the incidence of community-acquired pneumonia or CAP. Next, the study would like to identify the etiologies and the microbiota profile of hospitalized patients with the CAP. What is happening? Okay, previously. So actually, this is the workflow of the study. We perform culture and real-time PCR to confirm the etiologies. In addition, we conducted 16S sequencing to profile the microbiota from the sputum specimen. We divided the subject into 2 groups, mild, moderate, and severe, and we checked several variables contributing to the severe CAP, and we found that age is the only risk factor contributing to the severe CAP in the study. More of the patients are polymicrobial, followed by bacteria and virus as a single pathogen. Next, for the microbiota profiles, we found there is no statistically significant difference between the mild, moderate, and severe group. However, we can see the trend that in the severe group, the alpha diversity higher compared to the non-severe, and it is confirmed by the Spearman correlation in the heat map that on the severe group dominating by the pathogenic bacteria. Next. In conclusion, CAP in adult mostly caused by multiple pathogens. Patients older than 65 were 6 times more likely to have severe CAP than younger patients, and patients with severe CAP had higher alpha diversity but had more pathogens and fewer commensal bacteria. That's all, and thank you very much.
Sorry, before we proceed, can we show the time?
He's asking, okay, just hold on for a second. So you were asking whether or not you can see the time up here, the timer. So in this arena, we haven't been showing that. It should be shown, but you have used, they've completed their time. So now what will happen is you'll have 6 minutes. You'll have a minute to ask each of your questions, and I will time you when you're asking the question, and I will time her response if it occurs within a minute. And when you give your presentation, we will let you know at about a minute and a half whether you're over time or not on the next round. Okay, so for now, the floor is yours to challenge them.
Okay, thank you for the interesting presentation. We're wondering why didn't you include the control group? Because maybe the insignificant value that you got, maybe because you didn't include the control group? Maybe the mild group and also the severe one is not significant because they have similar maybe etiology or something like that. Thank you.
Thank you for the question. So first of all, we want to compare between the mild, moderate, and the severe ones because we would like to see the shifting when the patient got more severe pneumonia in patients. So that's the first point of the study, and we can see here it is the shifting between the mild, moderate, and the severe ones, and even though it is not significantly important because it is like when we want to really see the correlation, we need to observe the patients longer for the shifting of the microbiota. But in this case, we only took samples in one point, so only once at the hospital admissions. So that's why. But already since the beginning, we can see the shifting. So that's the point. Thank you.
Can you elaborate what shifting is that? Like maybe the microbiome profile?
Yeah. So the microbiome profile is already more diverse in patients with severe, and it is maybe at a glance it's not logical, but when we see the genus of the microbiome, we can really see the pathogens are more abundant in the patients with severe pneumonia.
And we're wondering, how do you trace and determine the cap? Do you trace that maybe the patient have visit hospital and they acquire the pneumonia from the hospital, something like that?
Oh, okay. No, a cap defined as a patient have the pneumonia outside of the hospital. So the pneumonia are divided into two groups: community acquired and hospital association. So a cap means that the patient, when they come to the hospital, they already have the pneumonia from the house. So it's made a community.
Oh. Okay. Yeah. Please show maybe the table too, because you said before that you want to assess the shifting. There's a table in your poster. Actually, you kind of combine the micropathogen profile between mild and severe. And why is that? If you want to assess shifting shifting value, why you combine? Why you do not separate those two groups?
From for the etiology, you mean? From the etiology
for
table?
the etiology.
Yeah, actually, so when a patient first come to the hospital, we did not severe, so we assess at the first time. We call it pneumonia severity index. So we assess the patient with severe they are. For the etiology, we haven't divided it yet. So for all the patient, we'll examine for to confirm what kind of the etiology in this patient.
So you mean for each group?
No, it's in general. So for the common practice for diagnosis in patients with community-acquired pneumonia, either they have mild, moderate, or severe, they will go the same procedure for the diagnostic. So that's why we combine this. And the main thing that we can see here, that most of the patients, half of the patients already have multiple pathogens in it. And actually some recent studies, for example, from Wooten et al. At 2019, it shows that when the multimicrobial contributes in patients with pneumonia, and then after that, also the other microbiome can really give some more advantage to the bacteria pathogens. So that's why we can see here the multiple pathogens is the dominant one, and also.
Okay, so here you include a lot of factors and everything, but to our understanding, you also included the genus, and we don't exactly understand what attribute of the genus that you find the correlation for. Like, why would you combine it? And it's not stated why like
Yeah.
what attribute and everything?
Yes. So for this, we combine all of the factors, but mainly we can see here from the severity index. So it's the PSI, and then we can see from the Spearman correlation which bacteria is actually significantly dominant in patients with severe pneumonia compared to the mild moderate. And then also we can see the one that significantly lower in patients with severe pneumonia compared to the mild moderate. We are planning to make a separate diagram for patients with mild moderate and the severe one, but it's not in the poster yet. Yes.
Okay, maybe this is last question. You categorize the mild moderate CAP as Psi 1, 2, 3, but.
Thank you so much. Very interesting. So that was a first round. So you have 1 minute to break and strategize, and then you will present. Sure. Okay. Let me know when you're ready.
Prognosis leading to reduced overall survival. And actually, mutated genes in primary tumors associated with positive and negative malignant acid tests has not been systematically investigated yet. So we want to address this knowledge gap. So our aim is to compare mutational profile in high-grade serous ovarian cancer, confirm positive acid test versus negative confirmed acid test. And we want also to compare mutational profile with patient with and without malignancy. And we're doing our research by extracting the DNA and doing library preparation and whole exome sequencing next. And we're doing our data analysis by establishing our in-house pipeline.
Yeah, this pipeline has already been benchmarked, and as such is already validated. Can you go to the next slide, please? So essentially, this is going to be our final. This is going to be our final mutational profiles. It's going to look like this. We have 8 samples in total so far, and we're in the works to generate this mutational profile. Essentially, what we're trying to do right now is to compare from different variant colors. Thank you.
Very good. Right on time. Excellent. Okay, the floor is yours for challenge. D41.
Okay, thank you for the interesting presentation. First of all, I would like to ask you about the normal that you mentioned as control in here. Do you mean it is like the normal cells of the patient, or it is like the normal people that you? Yeah.
So the normal frozen tissue were obtained from the same patients because we want to only call the somatic variants, and we want to like remove the germline variants. And the germline variants was acquired from the normal tissue of the patient. And yeah.
So for the whole exome sequencing data analysis pipeline, it's essentially like a tumor versus matched normal, and as such, we would be only interested in the somatic variants only. We use MUTAC2 for it, and we're trying to validate it using FreeBase as well. We call all the variants, and then we do our own somatic variant filtering.
So for in patients with already with cancer, how did you really divide the normal cells and the cancer cells? So because like the DNA probably despite that you cannot really see it under microscope, it may be already altered.
There are actually several methods to really compare the normal and the cancer. It's actually from the bioinformatics pipeline. We can see from we call it as a mutation allele frequency, because if it is like less likely to be called, and then it's probably normal. So yes, it is the reality. And during the data analysis on whole exome sequencing, we mostly do that kind of. Okay, thank you.
Okay, you have the result, all right? So. How do you anticipate your result if it is different from the benchmark? Because you still don't have the result here. When you then have the result, and then it is different with the benchmark from the already published study. Yeah, how you anticipating?
The thing about the paradigm of this research is that it's quite novel, in the sense that because we're trying to find. There's no published article so far related to this research paradigm, and as such, we don't exactly have a benchmark to really validate it. We do have it from the bioinformatics side, like pipeline stuff. The pipeline is already validated, but for the results, yeah, I suppose
it will be really novel. You know.
It will be really novel.
Oh, actually, when we read your poster, we also performed some library research. We see the literature, and we found a study by Simonian in 2020 in Nature Communication. Actually, they published 43 highly ranked genes with this kind of variants. That leads to the other question. Why did you also perform the whole exome when you can really actually do the targeted sequencing based on the previous research? Thank you.
Okay, so first of all, this study is novel. We want to assess any mutated genes that are associated with positive and negative acid tests that will be make the prognosis of patients become poorer. So why we're not doing targeted sequencing? Because we want to really assess wide range of exons to maybe we will find novel mutation that may give hints for this paradigm.
However, you only use 8 samples in here. Do you think it will be representative enough to the patients? And
okay, based on previous study, we have already systematically reviewed, and our current sample will be the biggest one, and we don't stop that. We don't stop at that point. We also want to validate our cohort with TCGA cohort, which already have 200 and more whole exome sequencing.
so, as you mentioned mentioned, you already see some systematic review about this. So, the study actually not novel.
It's novel in the sense of methods because they acquire the sample. Oh yeah, sorry. Previously they only do targeted sequencing, not whole exome sequencing. That's also the one that make our study much stronger than previous study. Yeah. Do you understand?
Okay. Yeah, but 8 patients with or without acid test, it is already combined, or 8 patients with acid test and 8 patients without acid test?
Okay, so the 8 patients includes the 5 patients with positive acid test and 3 patients with negative acid test.
And it will be sufficient for the.
In whole exome sequencing study, how many is many is still controversial. We try our best to do this initiative study. So hopefully in the future we will induce more bigger study to validate our study. Thank you.
Thank you. Very nice match. Very nice analysis. Okay, so we'll have give us a few minutes. Remain seated. We'll be compiling the scores, verifying them, and then posting them there. That was a good experience.
Negatif acid test sama yang positif acid test ini dibagi, dibandingkan. Iya, nanti dibagi mutational profile. Jadi nanti kita bisa. Iya, tapi berarti kan cuma punya 3 yang sampel acid test. Walaupun ya ada studi yang mikrobiota itu cuma 6 sama 6 juga ada, cuma kan ini dia 3 sama 5. Ya sudah, kita nanti sampel. Berarti kita salah, tadi kita pikir orang normal, makanya kita tadinya udah nulisin overcome ethical clearance kan.
Okay, so the winner of this match we declare is D41.
Thank you.
So so much. Congratulations to both of you. Great. Nice work. Very nice match. So take a look at where you are on the next round.
We have a lunch break for
Prepare
1
for your
hour.
next lunch break for 1 hour. Okay.
Start now.
Okay. Lunch break for an hour. Check out your next opponent. Go for it. Nice work. All right. Very nice. Well, very serious challenges. Serious challenges. Nice work. Excellent. Good job. Good job. Get ready for your next one. Get ready for your next one.
Pakai apa namanya, pakai page-nya Mac.
Tapi yang di drive kan yang upload gue.
Oh iya ada deh, yang di drive, yang upload yang D14. Bukan yang di sini di apa namanya, grup apa tim pribadi. Ya nggak bisa dong, harus dikirim.
Iya.
Dari pabrik tuh dikirim dulu ya.
Ini Mbak, break sebelum
Iya, 1 jam Pak.
Ya, nanti tidak boleh makannya di luar semua ya.
Oh, gitu. Nah itu Lina, yang 14 ini. Iya.
14.
Ini.
Iya, kan bisa kan admin mah. Yang mana ini?
Itu, dia 14 TPTS.
Nah itu,
Bisa nggak?
Tapi kan ini belum dirubah nih.
Boleh yang di sini.
Iya, iya. Belum dirubah. Coba lihat dulu. sama itu enggak konklusinya juga. Oh, ini salah. Konten yang diperbaiki. Gimana dok?
Ini aja kali ya? Ini belum.
Taruh ini aja deh. Gue pasang yang tadi. Yang tadi terus ke. Oh itu tinggal mindah-mindahin dong kan jadinya. Kemana ya? Gimana nih? Hah? Jadi pengalaman. Jadi gimana polling? Boleh di
Sebelah sini aja kakak. Soalnya ini agak settingannya. Silakan. Mau yang ngomong ya? Gue mau baca itu soalnya. Iya.
Cek 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 satu cek satu cek satu cek satu cek 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 tes tes satu cek 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 tes 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 cek 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 cek
Say start, you can start. Okay. Hold on a second. Let me see. And yes, you can start.
Okay, hello everyone. Welcome to our project. So as you can see here, that antibiotic resistance increase every years, and we need the new drug discovery. In our previous study, we found the microbial natural compound, microbial natural product from mangrove api-api in Yogyakarta, and based on the 16S, it's identified close belong to Racemicystis. So the aim of our study consists of three. First is the antimicrobial activity of the strain against the microbial pathogen, and the second one is identify the biosynthetic gene cluster, and the last is identify the biosynthetic pathway. So the method consists of four. So we starting with the cultivation and extraction, and we continue with antimicrobial assay against the pathogen, and the third is the metabolomic analyzing using LCMS, and the last is the genome analysis. We use the Illumina. Then next slide, please. And this is the mock-up that our result, and our result highlight that the strain have the antimicrobial activity, especially against the Candida albicans. And the second one is the metabolomic and genomic approach unveil the biosynthetic mechanism of the strain to produce antibiotic compound. Thank you.
Okay, from the result, we identify that the strain had the the highest activity against Candida albicans, and from that result, we use the strain to analyze the metabolome and also the whole genome sequence. And from that, we integrate both of the result to get the biosynthetic pathway and analyze which compound was expressed. That's all.
Okay, the time is yes. The time is up, yeah? Okay, and then ready? With the question. Your turn. Start.
So how do you know that they actually have this? Because you do the assay against candidiasis, but when it comes to the genomic structure, what does it say anything about they have the antimicrobial activity? Which genes? How do you decide that?
Maybe I can show you that this is the heat map of our study. From this result, this is the pathogenic bacteria, and it showed that among these, the highest activity of this strain show against the candidiasis. While the biosynthetic gene cluster, we had not yet analyzed it, but from the whole genome sequencing, we will identify the biosynthetic gene cluster that had been in the strain and what type of gene cluster that the strain had. Then we try to analyze, do the metabolome correlate with the PGC that we got during the analysis.
Because the candidiasis is what's the mechanism of how they kill the candidiasis? What kind of products of this gene that actually kills?
Yes. What we mean about biosynthetic mechanism is how this strain producing, not the mechanism of action of the molecule itself, but the biosynthetic process or the metabolomic process to produce certain compound, not the mechanism of action of the compound against the pathogenic bacteria.
Yeah, because from this data, it's not really clear whether you can put this together. I mean, connecting the metabolomics and then the functional assay.
Yes.
How do you, which area to prove that this cluster actually killing, or which cluster is just basically passengers?
No, what we mean is we analyze what PGC that is available in the whole genome sequence, and from that we try to predict what kind of compound of the PGC will express, and then we integrate or we correlate with the metabolome that has been expressed and identify using the LCMS-MS.
Okay, does it actually correlate with the effectiveness of killing? Because from this mock-up, I don't know which one said the proof that actually they're killing and something.
Oh yes, this is the heat map. It is not related to the BGC itself, but it is our preliminary study, which analyze what do the extract have the activity against several types of pathogenic bacteria, and from this result we choose one of the isolate, which is racemicitis, and then we analyze both of the extract and also the whole genome sequence. It is just from our first founding that leads to the other things.
What is generalizability of this finding? Because it's only specific to candidiasis, or it could be extended to maybe antibiotic resistance bacteria? How generalizable this finding?
Sorry, can you repeat?
Yeah,
the
can you
question?
actually predict they have the same antibiotic? The microbes are so many, right? Not just candidiasis. How about against some other bacteria, say antibiotic resistance bacteria?
We also tested the extract against several other pathogenic bacteria, such as Staphylococcus aureus, Escherichia coli, and the other kind of pathogenic bacteria. But this strain had the highest activity against candidiasis, because it showed that the MIC is the lowest compared to the other type of extract that we had.
Last question,
Can you actually, because there's some specificity, it looks like higher toxicity against candidiasis, but not the other one. Which one from this data show that the genetic segment actually responsible to killing candidiasis, but not the Staphylococcus?
We had not yet identified which one of the BGC was responsible, but we try to integrate both of the data in the next session, because we're not yet finished our study yet.
Okay, thank you. Okay, so we have a next turn. Hold on. Get ready, start it.
Oh, not yet. The
Oh?
the slide.
The slide.
Yeah,
Okay.
the slide.
Please. Ready? Okay, start.
Yeah, Bismillahirrahmanirrahim. So let us present our work in detection of DNA methylation from buccal swabs using NGS to study stunting. So next slide. We know about 20% of Indonesians children have stunted phenotypes, but in Pandeglang it's extremely high, around 30%. And we know chronic malnutrition of the mothers actually is a major risk of stunting. And currently, the proposed mechanism is lack of metal donor because of insufficient intake in a nutrient that actually may affect the epigenetic programming of the babies and the children. So we tried to identify this marker, this epigenetic marker that associated with stunting using buccal DNA, because most studies using blood, but now we're going to use a specimen that's easier to collect. And the method we use is NGS that can detect actually the native DNA methylation in this buccal DNA. Next slide, please. So we start with collecting 200 children in Pandeglang, and we choose the top 12 samples, 6 from normal and 6 stunted children from this buccal DNA. And then we use this NGS to look for differences in the methylation between this normal and also with the stunted children using this NGS. And the data shows, next slide please. Apparently, there are some differences. If you look at the arrows, the red arrows, the top one is normal, the bottom one is stunting. The green indicate the extent of methylation. If you look at the stunting, it seems to have less numbers of methylation, which may be consistent with the idea that a lack of nutrient. So we choose the IGF H19. H19 because they've been published previously, and the similar pattern has been shown with also other gene locus that maybe point out that this method is useful, and we're going to plan to use more samples later on. Okay, so we're going to plan to do analysis in further more children, and also to follow up children for the next year, whether we can see a consistent changes in this methylation data.
Thank you. Yeah, start the question.
Maybe you.
Ready? Time up. Ready? Okay, start.
Okay, maybe you can collaborate more about the ages of the subject of the study, because in the poster I read that the diversity of the age is pretty much not very diverse from children to adult.
Oh, okay. So we choose the age is about between 0 to 5 years old, and out of this 6, because we try to arrange quite similar, so under 2 and also 3 years and 3 years and older. So actually about 50-50 in both stunting and also with normal. So currently we try to do some more analysis to stratify according to age and also according to sex. But right now we still focus on the phenotype first, the outcome is stunted in these children, the group of children.
And about the group of the severity of the sample, is it have been categorized specifically because the severity of the stunting is pretty much diverse?
Right. So we try to have actually a couple of category, stunted and severe stunted, but for this particular ones we don't do that yet, because we try to make sure the technique is actually working, because this actually the first technique has been done in the world to use this adaptive sampling to look at the native methylation, where we don't have to do some chemical conversion of the DNA. So when the technique is consistent, we can tell from the differences of the normal and the stunting. That's in pointing out some interesting features. Of course, later on when we have more data, because right now we start first with the top 6 first with the DNA is very good. And then we work later with more samples with different category to make sure the phenotype still hold true, but the difference we can see is still consistent.
But I see from the methodology section that the quality of the DNA itself from 230 it reduced to only 12 that had been qualified for further analysis. Would it be a problem for the representativeness of the sample to the whole subject?
So we attempt to make sure that the technology of NGS requires high amount of DNA. So of course, when we choose this 6, is to make sure they meet the minimal standard. So once the minimum standard is done, because that means we already have the algorithm for the bioinformatics analysis, we figure out the computation, and then we're going to move to the next amount of DNA. So this is actually the first time that we can do it in the world, because if you start with a less amount of DNA, and then if it's something failing, we don't know if it's got DNA or because of the biology, right?
Yes, but is it possible to redo or optimize the extraction method to get the better quality of the DNA sample?
Sure, definitely. So once we know the minimal amount, then we can make sure that look at the rest of the samples that meet this number of samples. And the good thing about this cohort, we know exactly where they are. So if it's necessary, we can go back and re-sample this of these children if it's necessary, because we have actually a good collaboration with the village in Pandeglang.
Yes, because I thought that it will be better if you had more data to get the more representative result compared to just use 12 that is met the data quality but not the representativeness of the whole sample.
Right, this is going to be a next series of papers, because definitely this we focus on the technicalities first, just to make sure that our computation is okay. And we also look at the different genetic loci has been shown previously that have a different methylation between sufficient and insufficient nutritions. So this actually was very encouraging for us to move to more number of samples.
Is it possible to show the visualization, not all the result is just the highlighted section, to you do another statistical analysis to compare it between the normal and stunting sample?
Right. So the bioinformatics staff that has been doing this, she's still doing it, but she reported to me that at least from these 5 genes have a similar pattern like this what I presented here. But of course, later on we have to do calculation because in instance we have actually 30,000 sites of methylation that we can look, not just these 5. But this 5 is represented visually that we can look very easy, because to look at 30,000 is not that easy to show the differences. So we focus on those that have actually has been published previously and have a very interesting mechanism how they explain why these children got stunted.
Yes, maybe you can really correlate more about the normal and stunting between both of the graph in the above and below.
Right. So if you look at from this low side, there seems to be 10 differences that the stunting has actually less numbers of methylation, which is consistent with the idea that when the methyl donor is insufficient because of the nutrient, then you have less numbers of methylated nucleotides in the genome.
Is it possible to identify which gene that is correlated with what kind of factors
for example,
that affect
in these
the
areas,
stunting subject?
when you look at this negative data,
Okay, thank you. Yeah, congratulations for the both teams. Okay. Okay. Congratulations and give greet for both teams. Yeah. Thank you. Thank you. Thank you. Selamat. Selamat. Thank you. Ya. Apa? Langsung masuk berikutnya. Atau ada dipanggil dulu atau gimana?
Untuk yang berikutnya for next session is D30 and D49. Please.
Are they here? Sure you know each other, right? Okay. Would you mind to make a quick introduction? You first. Yeah.
Thank you so much for the opportunity. My name is Yeni. I'm from National Research and Innovation Center, Indonesia.
My name is Fikriya Hanum Humairo. We came from the same institution.
My name is Indira. I'm from InBio Indonesia.
Okay, my name is Fahrul Rizal Mahindra. I'm from IPB University and InBio Indonesia.
Okay. Yeah. So we want to have this session. Do you have any question about the role? No. You're clear, right? Yeah. Okay. So we want to start. You know the judges here. Okay. Pak Iqbal and Bu Ati, and we have the technical team as well at the back. Okay, so I'm Ahmad Hidayat. I'll be the referee for this session. So first of all, we want to have a toast. So you will choose which one gonna be the presenter or the defender. Okay. So please choose whether this side or this side.
Head.
Which one the head? This one, eh?
Yeah.
So you are this one, yeah? So they are here, okay? Okay. Oh.
Okay,
Head, yeah? Okay, so you are the first. You choose. You want the presenter or defender? Hmm?
We are going to first
Present first. Okay, please. You have the 2-minute presentation. Okay.
Actually, we need 2 mics.
You need 2 mics. Okay. Please get ready. The slide is up. Okay. Ready?
Start.
Start.
Okay. Now. Good day, my lovely audience. I would like to tell you about our story. We went to the Sulawesi, the eastern part of Indonesia, to study the most expensive food in the world. Now, maybe some of you are asking, do we really have that, or why do you have to go thousands of kilometers to study that?
Well, yes, we have ebony, an endemic of Sulawesi. Ebony is one of the most expensive wood in the world because of its strength and durability. However, based on IUCN, ebony is listed as a vulnerable species and one of them because of the plant disease.
Thus, we came with this idea. If we can understand the microbial diversity, in this case bacteria, from the asymptomatic and symptomatic leaves of ebony leaves, we can formulate the biocontrol against this pathogen causing the disease.
Next. So first of all, we thought that the symptomatic leaves may have more bacterial pathogen. However, we found a very interesting result where the symptomatic one actually have more beneficial bacterial community. As you can see in Spingomonas, Methylobacterium and Bacillus is actually higher and abundant in symptomatic plant. So those bacteria are highly reported to have antipathogen activity. So how can be happen. So this is one strategy of the plant to survive from the pathogen or stress condition by recruiting some of the beneficial bacteria for survive in cold conditions. So it is very nice to have those isolate the bacteria and then formulate them as a biocontrol agent. Thank you. We are Team D49.
Thank you, and we are open for discussion.
Still have few minutes, second slide.
Really?
Okay. Okay. Get ready with the questions. Timer. Okay.
Start now.
Okay, thank you. I have the first questions.
So yeah, we see that from your poster and in this your result that you didn't use control leaf in this research. So how can you compare and show the significance of your result asymptomatic leaf and symptomatic leaf compared to the control? You didn't compare it.
Yeah, well, actually there's no control because we just compare this between healthy leaf and not healthy leaf. So I think the healthy leaf will be a control for this kind of study. So this is a descriptive study. So we want to compare the bacterial diversity from those leaves. So there's no control. If you say control, then it is the asymptomatic leaves.
This asymptomatic leaf and symptomatic leaf, is it healthy or disease or has disease?
So when you say that it's symptomatic leaves, it means that it has a potential disease. But we cannot call them as disease because we still don't know about what kind of disease in here. But it has symptoms like that. So we cannot say that disease or not disease, but symptomatic and asymptomatic.
Okay, I have this second questions. You state that several microbes have the potential to biocontrol activity to maybe in the pathogens in the street, and then and you validate with the reference, right? Reference. Okay. I have a question. Why you not use the maybe analysis to bioinformatic or the database to alignment of the enzyme, maybe the enzyme nomenclature, AC or analysis, GOG analysis or seed analysis to align your data to the database, maybe using the BLAST analysis to analyze the activity of bioprocess of in the bacteria.
Yes, thank you. So we just use the 16S rRNA gene. This is a basic gene for identify the bacterial identification, but we don't use any kind of gene. Speak the gene and conserve for bacteria. However, we are in progressing by isolating bacteria, and we found Spingomonas and Bacillus as well in our culture isolation, and we already tested with several of fungi, fungal pathogen, and it works very well as a biocontrol agent.
So have you tested this Spingomonas, methyl, and etc.? Have you tested it if it can act as the biocontrol agent of ebony?
Not in ebony, but some fungal pathogen, because we still don't know what is the cause of the disease in the ebony. So we put this as a fungal pathogen, for example, like Fusarium and other types of the pathogen. I forget the name, but we already validate that some of this, like Bacillus and Spingomonas strain, has a very effective biocontrol activity against this kind of fungal pathogen. However, in here we also try to see the metagenomic of the fungal diversity in those leaves as well, and we found also some of those fungal from this leaf sample.
Okay, so it still have a potential that the ebony may have disease other than the fungal, right? So how much you can assure that your finding is significant for biocontrol agent, because we afraid that you might overclaim it. You have stated that we could isolate the bacteria and use them as natural biocontrol.
Okay, thank you so much. So we have some of the fungal that we are already isolated from the leaves, and then the next step is actually we want to do the postulate coach to the plant. So we want to try to inoculate those fungal, and we want to see if it causes the same symptoms that we found in the natural plants. So that's how we confirm that.
And we still have time?
Yes, yes.
How about the statistical analysis that you use? Because in this graphic, we see that the relative abundance is quite low, because compared to the relative abundance in this left graphic, the top value is 1. Meanwhile, in this is only 0.04. So I think it's not quite significant.
Okay, so actually we don't do any significant analysis because we only have like one replication of the sample in each analysis. But the idea is we try to composite the sample from that we take from some location of sampling site, and then we combine the samples, and then we do the analysis with the sample. And so why this is 1 and why this is 0.04? Because 1 is because we put all of the bacteria here, but in here it's only one kind of genus. For example, like Spingomonas, Methylobius, Jatropy habitans, and etc. So that the value is not until 1, the abundance. Yeah.
Yeah, but still, you cannot state that this finding is significant, right?
Yes, yeah. Of course, yeah, I agree with that because this is only one of our preliminary study.
Time's up. Okay. Yeah. That's it for this group, and we'll take turn for D3630. Yeah. Get ready for the slide.
Check. What?
Yeah, can you fix the microphone?
You want to use
Can
this?
you make sure the microphone is right?
Okay.
This one.
Referee, if I may ask, do we have any pointer to help? Probably.
Yeah.
Because sometimes maybe we
Do we have a pointer? Do you need it? The pointer?
No, actually.
No? Okay. Okay. Okay, the slide is up. Then, are you ready?
Yeah.
Timer?
Okay.
Okay. Can we start now?
Start.
Good afternoon, everyone. So here's our topic. It's about bioinformatic analysis of microRNA mediated regulation in their potential as therapeutic targets for lung cancer. Next, please. So based on global cancer data, it has known that lung cancer has become the most fatal cancer. The previous study has shown that the current cancer treatments such as chemotherapy and radiation still face high mortality rate. Meanwhile, in the previous study, the microRNA can act as tumor suppressor. With the help of argonarotein, the microRNA can degrade the mRNA. So this research aimed to identify the most potential microRNA as the novel therapeutic approach for lung cancer in stage 1 until 4. This research utilized the microRNA data from the CGA database and analyzed it with informatic approach. Next. So from heat map and ANOVA analysis, it showed that significantly different expression level between normal and cancer sample. And the volcano plot split the microRNA based on the expression profile. And the table here show the list of the most potential tumor suppressor microRNA in stage 1 until 4 and its target gene. Next. Next. The selected microRNA have been evaluated with ROC analysis. Meanwhile, the target genes shown to be involved in the pathway and biological process of cancer. The molecular docking show highly binding affinity between the miRNA-mRNA duplex and argonaute protein, and this is the visualization of the docking. So the conclusion of our research is these 4 microRNAs have potential to be developed as therapeutic treatment for lung cancer in stage 1 until 4, but the further validation with in vitro and in vivo still needed. I think that's all from us. Thank you.
Thank you.
Okay. Timer ready.
Okay. You can start now.
So I have one question in here because this is like a data mining, right? So you choose the TCGA database for your source of data. So do you have any other kinds of data? And if yes, why you don't like combine from the other database as well? Why only you choose one database?
Okay, good questions. We use the TCGA analysis because the TCGA database is very comprehensive data that have the data of the mutations, data of the expressions, and the other platforms use it. And there is the several data orders in the TCGA, like the GEO database, but we use the TCGA because this TCGA is the complicated and have the all data about the sample and maybe the stadiums and the pathway and the clinical trial and clinical activity of the samples.
So actually we want to make sure the data and the database that we use is already comprehensive enough to do the analysis.
Okay. I'm still confused. Why do you have to choose the complicated one than the simple one? Because your study is very complicated as well. What I want to see, ask, I see in your poster that you involve 518 tumor patients and 46 normal patients with lung disease. From what I know, there's nothing normal in patients, especially lung cancer, who has the lung cancer. Can you please elaborate? Because I'm questioning your eligibility criteria of your study.
So so in the TCGA database, it already divided the patient between the normal and cancer cell. So there is a value, the log fold change that it is used as the predictor between the normal and cancer cell, and the more higher or the more lower of the log fold change value, it means that the stage of the cancer is more severe. So it's like that. So there is the value that used as the standard to differentiate between the normal and cancer cell.
I think you can answer whether there's a to answer this question.
Yeah. Do you have any reference with the standard value?
Yeah. Can you go back to the again? Next. Yeah. So here, like in the in
3
this,
minutes.
in this table, the log flow change. This is the lower of the log FG value means the microRNA in the lung cancer is more downregulated. So it's like this is the example of the value. But the reference paper we cannot show it in here, but you can find it on the CGI database. It has already a comprehensive reference of each of the sample.
Actually, I'm still confused about the data mining. I mean, like, how can you normalize this data? I mean, this data is come from like different sequencing platform or microarrays and sample preservation or sampling. How can you normalize it? Like, how it is like your data really reliable?
Okay. I see before about the how to the project sampling. This is the you can see at the GDA the data of microRNA established and apa yang didapat and get from the sequencing and apa itu? And we use the normalized data because we not use the normalized data because this data have the source the one project from the one projects. It's not the other project, but it's one project from the one projects, and the projects is divided in several populations, normal cancer and several community like the race, black race people, white people, and Asian people, and we normalize the data. After we get the data from TCGA, we use
1
the
minute.
scaling data, normalized data using the RStudio and other page kits like the DEJ analysis page kits and RStudio to normalize the data.
If you said that it is from one project, so this data cannot be generalized.
Yes, this is from the one project.
One project only.
One project.
So can it be generalized? So can your result be can be generalized to like the whole population of the?
Okay, the project is ada beberapa. Okay, the project has several data from population. The black people and white people and the Asian people, and I think this is a very apa ya bisa coverage the all global populations.
Okay, I think you better to show it in your figures because we didn't see the white people, black people in your figures.
Okay, time's up. Thank you.
Pernah coba?
Karena nggak stabil.
Kecil sekali. Ada satu lab yang punya electron microscope untuk bisa isolate itu.
Saya di Cina, Pak.
Oh, dia di Cina? Enggak, ada di sini kok. Di Serpong. Lab swasta dan buat. Nanti saya bicara sekarang. Tapi Anda bedain antara healthy dan unhealthy itu Anda nggak anggap itu sebagai kontrol? Enggak ya?
Okay, we get the total scores.
Okay. Yay! Okay, you can congratulate.
Thank you.
Thank you.
Okay.
Okay.
Photo session? How many groups do we have? How many rounds?
3 rounds before break.
3 rounds before break. Okay.
Yes.
3 rounds. 3 rounds. You want a break? You need a break? Okay. Okay. So next round, yeah?
Yeah. Iya. Silakan. 13 sebelah sini. Kita mendaftar dulu.
Nanti diskon nggak masalah kan tadi aku
Poinnya kamu itu.
Enggak kok. Kita salah ini. Bukan. Untuk yang 45.
Iya, 45 ini.
Oke.
Oke deh. Udah benar grupnya? 13, Anda? 45 udah benar ya? Okay, good afternoon everyone. So we want to have this Dojo session. Do you want to introduce yourself? Start from you.
Hi everyone, my name is Edward.
Yeah, I'm Hera. I'm from Sun Institute for Development.
Okay, the host.
The similar name, Hera. Okay, I'm from Brin.
And you, Pak?
and I'm ready.
Rin. From Brin.
Yes.
Okay, so we want to have this session. First of all, let me introduce myself. I'm Ahmad Hidayat, and we have two judges, Pak Iqbal and Bu Ati at the back, and we have the technical team as well to make a timer working. Okay, so we want to have a toast so that we can choose which one going to be the presenter and who's going to be the defender. Yeah.
Yeah.
Who's win the toast, then you be able to choose which one your turn. So which one do you want to choose? This one?
Head. We just
Head?
Yes.
And you
Number
tell?
10.
Is okay?
Yes, that's okay.
Alright. Well, it's up. Oh.
Head.
Head. Okay. So you choose.
Yes.
Present first.
No,
Okay. Huh?
uh,
Oh, they
the-
present.
yes.
Okay. All right. Okay. Make it the slide up for this group. D13.
Pakai poster kan?
Ganti, Mbak. Ini 3.
13.
Aku 13.
It's 13. Kita berapa gitu? Tahun ini.
Yes. All right.
Alright.
Yes, this is right.
Make it the slideshow first, or you want to keep this way?
Not this one. Yeah. Okay. Because it's in the PDF.
Okay.
Yeah.
Timer ready?
Okay. We can start now.
Bismillahirrahmanirrahim. All right, we did 13. That our aims, the research is to provide, to ensure that all the goodness from the stream pond to the plate. So we found that there's a recent study said that food preservative or disinfectant have an unforeseen impact in the gut microbiome. So we just want to make sure that your stream is good for your health, and we also want to have like a basic foundation for further research, improving the processing quality of the stream, and also for the good health in the future. All right, so we have like a stream in the Serang Pond in Banten. Then we also have like a mini plant facility to frozen the stream. So we have like a control as okay. So the control is sorry. So we have a CPC 0.05 and 1%, and the control is chlorine because this is usually used in the stream industry. And then the result is, next please. Yes, there's a significant difference in the sensory after washing for the stream with the CPCs and also the fat and protein content. But in the graph plant, the pond water also have a different kind of bacteria in the cold storage after 14 days. And the CPC have the effect of the fat and protein content, and Picocorum sp and the Nanochloropis gaditama is the most abundant species in the stream, and the CPC is really suppressing the Salmonella and Vibrio. I think that's all.
Okay. Get ready for the question. Okay. Timer.
Okay, we can start now. Yeah, thank you for the presentation. But we have a concern to your study, but that there is no clear information both on your slide and your presentation on your poster regarding the sample size. It seems like you can only use the just only one stream. We don't know.
Okay. All right. Yeah. We don't have. Yeah, we don't include that, but the stream is came from the ponds in the Serang. We take 100 kg and we process in the processing plant around 100 kg, but we block it in the block stream 500 kg, and based on SNE 1996, if you want to take sample for seafood production, the product is if the sample is less than 100 grams, you can take like 100 grams. So yeah, for the information for the microbial community, we take 100 gram and then mix it and take for the DNA.
For the protein result, if the committee can zoom it, your result doesn't make sense. You conclude the 0.1 CPC actually reduce the protein content, but the 0.05 CPC actually increase the protein content. So like, how do you make sense of this data?
Yeah, this is also our questions, and we're still looking for what the mechanism behind that. But we take the sample like 3 times with the 2 times replication production, so the data is valid. But why is that happen? The mechanism that we still have to find out.
Also, some of your figure here have statistical analysis, right? Like significance and not significance.
Yes.
The rest, however, like figure 2, moisture and ash, doesn't have statistical significance. It seems like you're pre-selecting, showing some are statistically significant and some are not.
No, we have like a graph plan Prism 8, 9 actually. So they will appear automatically if there's significant difference. When they not appear, that means it's not statistically different.
And following up on that, so there's a lot of statistical analysis here. How do you ensure that some of this significant result is not only by chance? Because I'm pretty sure there's at least 30 to 40 multiple comparison here.
Which one do you mean?
Almost everything, I guess, right? Because when you do a multiple comparison, there's a random chance 0.05% where it just be significant by chance. How do you ensure that doesn't happen in your significant result?
We use the Taki following measurement for the multi-compare. So the non-significant is less than or bigger than 0.5. 1 star significance 0.0299.0.109, So the second star, like more significantly different to the one star. It starts from 0.002229, and the third star, yeah.
Yeah. So your significant level is still set at 0.05, right?
Yes, for the not significant. But when you do Taki, that's automatically counted. Yeah.
Okay. Also, how do you explain figure 4? So you mentioned about in figure 4, there's an explanation, like there's a difference in bacteria in pond water and shrimp after the storage, but for the fresh shrimp, like there's a different group of bacteria.
This is not the fresh shrimp. This is the SO. It's like the pond water. So this is like the media the shrimp is living. So mostly it's the protobacteria and bacteroidota. But when it was and after the washing and storage, it also have like a protobacteria, but then Firmicutes take over the bacteriidae.
Yeah, exactly. That's my question. Like, how do you explain the difference? It's not which bacteria.
Just because of the CPC. I think the CPC is selecting the have an impact of the different kind of the phylum or genus. That's still we have to discuss it in the discussion stage. Yeah, but that's because of the different kind of CPCs and the antibacterial things in the process.
Okay. You also mentioned your second conclusion, which is the Picochlorum and Anochloropsis is the predominant bacteria in the shrimp. Yes.
Yes.
But these are algae. These are not bacteria. Is there cross contamination here? So you're identifying algae as a bacteria in the shrimp.
This is based on the species that. It's not cross contamination, I think, but maybe because there's also have the bacteria that live in the algae. So I think that's identified as this one. Is this a further analysis? So yeah.
But bacteria and algae are two different class of organism, right? And
no,
your
I mean like,
objective was to look at the microbiota of the shrimp.
Yeah, that's why we also like asking this. Okay, thank you.
Okay. And the next one. Get the slide ready. Ready? Timer.
Hi, I'm Edward. With my partner here, Hera, we'll be looking at side effects or reactogenicity of COVID-19 vaccination among pregnant and non-pregnant women. So as we know, COVID-19 is associated with poorer health outcomes, both for pregnant women and also their babies. And therefore, vaccination remains a crucial public health tool. It's important, therefore, to understand how different COVID-19 vaccine type might have different side effects too. However, right now, what we've seen is that most studies came from developed countries using mRNA or vector-based vaccine. So we aim to fulfill this knowledge gap by providing a real-world COVID-19 vaccine reactogenicity assessment among pregnant and non-pregnant women in Indonesia. So, as mentioned in our title, we enrolled a prospective cohort in Lombok from July 2022 to October 2023, where we assess vaccinated women from the day after the vaccination and up to 7 days after their vaccination using WhatsApp, phone call, and home visit.
Yeah, and in our study, almost 85% of our participants reported any symptoms of adverse reaction within 7 days after they got the vaccination, which nearly 13% for inactivated vaccine and 72% for mRNA or mRNA vector vaccine for any local and systemic reaction such as the injection site, headache, and fever. And our study found that the lower reactogenicity were experienced by the pregnant woman compared to non-pregnant women. So our study suggesting that the potentially better acceptability of inactivated vaccine compared to mRNA or vector vaccine based.
Thank you.
Okay, get ready with the question.
We can start now.
Okay, thank you. This is very interesting. I want to ask from the title. So you mentioned you aims want to see the side effect, but there is no mention in your paper here. So for my did you check this activity of its vaccine? So it's not the side effect, but it's the how the how to say like the activity of the vaccine for the pregnant and non-pregnant woman.
By activity, you mean whether or not it's protecting the pregnant women? Yes, we check it, but it's sort of out of the scope of this paper because we only want to look at reactivity. There's a separate paper that actually look at breakthrough infection.
Okay, for the second one, in your title you use Indonesia, but you only take the sample in Lombok. That's right. Why you want to take sample in Lombok, but you want to describe this is what happened in Indonesia?
Yeah, so the reason why we can generalize it from Lombok to Indonesia is because the vaccine type in Lombok is the same with the rest of the Indonesian. It's the same, which is mRNA inactivated vaccine and vector-based vaccine. That's why we can generalize it to Indonesian, because we are using Lombok have the same national guideline like set by the MOH and by Indonesian OBGYN Society.
Okay, next, is the study pre-registered or not? If not, why?
It's not pre-registered because that's usually a common standard for an RCT. We are doing cohort, so we're not obligated to pre-register this study. This isn't a RCT trial.
And what is measurement that you use to minimize the bias of your research?
So in terms of minimizing the bias, we do a triple, as I mentioned in the method, we use 3 methods, which is through WhatsApp, through phone call, and through home visit to ascertain this assessment of reactivity, whether or not they do experience the side effect. Obviously, having these 3 measures increase our confidence to ensure the bias is much more minimal rather than one method.
How about the memory bias of the respondents?
Yeah, so we are tracking them every day during their 7 days. We're not tracking them at the last day of the 7 days. So I would argue they wouldn't forget their symptoms exactly at this D-Day, right? Because this is like, we're asking them at the D-Day.
Okay, so do you think that your sample size is good enough, reliable enough?
Yeah, so these are 2,000 strong sample. We don't necessarily calculate it like the minimum sample size, but at 2,000, these are pretty big study. So we are quite confident on the minimum sample size.
So for the figure 1, so there is a scale here.
Yes.
So in your expertise, this is 75 or 30 high symptoms for the pregnant woman. It's very dangerous or something like what in your scale?
That's up to, generally in terms of the severity, we did ask the individual how severe they feel the symptoms. So it can be mild, it can be moderate, it can be very severe. But we as the, as the investigator, we don't prejudge like whether or not these symptoms is severe. It's supposed to be self-reported outcomes from the from the vaccinated women itself. So there's a separate graph for that, but we didn't show here. But yeah, so we don't necessarily judge whether or not this is severe. It comes from the self-reported outcome from the subjects.
Okay, in some cases, there's mRNA and the vector vaccine. It's like a spike up in the four times 24. What do you think this can happen?
Yeah, so for this, the spike comes from the headache. We're not really essentially sure. It's only on the down where there is a spike happening. We're not necessarily sure if there's a biological explanation because we also didn't see this spike too with the inactivated vaccine. So this is something that need to be further figured out.
So what do you mean by others?
Others.
Others in your.
Others is other side effects that are less common. So the less common one are explicitly stated. So fever, headache, fatigue, pain at injection, and muscle ache. Some people might have diarrhea. Some people might have any other adverse effect. Those will be lumped together into the others category.
So for this scale, for any condition, there which one is dangerous for pregnant woman? If
again, it depends on the women feeling the symptoms directly and the clinician, right? These are common side effects. If it goes to the point where it's interfering the women's symptoms, they'll go to the healthcare facility.
Okay. Saya mau klarifikasi pertanyaan Anda tentang pre-registration ya. Sebenarnya itu harus diregister. Ada peraturan Menteri Kesehatan tentang observational study, registry study. Itu ada PMK-nya. Jadi, tapi pertanyaan Anda mungkin registrasi di clinicaltrials.gov atau WHO, ICTR atau yang lain.
Ya, di mana yang ada standar Indonesia. Saya nggak familiar sama.
Oh, Anda nggak familiar dengan regulationnya.
Kalau ethic clearance memang sih, tapi kalau pre-register setahu saya emang
ethic clearance-nya ada kan?
Ada.
Ethic clearance-nya ada, tapi sebenarnya Permenkes tentang registry study itu ada.
Oh, siap.
Di dulu di Bankes tadi.
Siap. Makasih.
Oke.
Thank you. Makasih.
Udah break atau belum? Belum, kayaknya 2 lagi ya?
2 lagi.
Oke, makasih. Ya.
Dari tadi.
Okay, before we start, do you want to introduce yourself?
Hello, Pak Ahmad, and everyone. We are from D31. My name is Nuril Maulidia Mawfiro.
And my name is Haryo Jaksono, and we're from
D31.
Be honest.
Yes.
Please.
Terima kasih. Thank you. We are Utomo and Utomo. Ahmad Utomo, Chandra Prastya Utomo. So we are from YRC. Thank you.
Okay. Yeah. This afternoon we will have a session. Are you clear with the regulation? We're good. Really? Are you good? Okay. So we want to have a toast. So the winner of the toast, we choose. Do you want to be a presenter or the defender? Okay. We have our judges here and the timer to help us managing this session. Okay. Which one do you want to choose? Head. Okay. You are the tail, yeah. Okay. So, do you want to choose?
Yeah.
Listen. You present. Okay. They will present.
Okay.
Get the slide ready.
Okay, we can start now.
Hello everyone, we're from D31 team. We will be presenting our project with the title Genomic Profile in High-Grade Serous Ovarian Cancer Primary Tumors associated with malignant ascites and tumor metastasis. Next. So our background to study this project is highly about high-grade serous ovarian cancer that predominantly spreads through the peritoneal cavity involving ascites during malignancy. And this tumor cell that accumulate in ascites has been associated with a poorer prognosis, which then leading to reduced overall survival. And actually, mutated genes in primary tumors associated with positive and negative malignant ascites has not been systematically investigated. Therefore, we want to address this knowledge gap. Our aim is to compare mutational profile in high-grade serous patients with confirmed positive ascites versus those with negative confirmed ascites, and also compare mutational profile with and without malignancy. And we're doing this project by by extracting the DNA and conduct a whole exome sequencing, and we're doing the data analysis by using our established in-house pipeline.
Okay, so the whole exome sequencing data analysis pipeline has already been benchmarked and validated using external open source data. And can we go to the next slide, please? Essentially, we're going to do a lot of post-processing, and this is going to be what the mutational profiles would look like. So on the left, we can see our samples, our patients, and essentially we're still conducting our analysis, but this is going to be our the way we are going to visualize the mutational profiles of those with positive and negative ascites.
Okay, so we have a Q&A session. Yes. The timer is ready. Okay, start.
Yeah, thank you. So the choice of having the source of samples, when you say the frozen, it's actually from the primary tumors.
Yes.
Okay. So the idea is to find differences. Okay. And any clues whether the difference has been found?
Like for this theme of study, there has been any study conducted previously to compare the primary tumor that associated with positive and negative malignancy in ascites.
Okay, because they are all historically are serous cancer, ovarian cancers, right?
Yes.
So I would expect majority should be the same anyway. How would you, because is there any hypothesis that which are the differences lie in what kind of genes?
Oh yes, there is a previous study that came from people who working group which analyze the TCGA database. They suspect that the cell adhesion pathway might involve in during this malignancy of the ovarian cancers, to be specific. So we suspect we will find some kind of after we study functionally the mutated genes that we will find, we will expect we will find like gene that function in cell adhesion.
Yeah, I was actually just to make sure that you were doing on the right track, rather than doing the whole exome sequencing. Start with targeted first, just to make sure you actually see some difference in the published literature, right? Yeah. Have you found some clues on those kind of genes that have been published already? I mean, jumping to exome sequencing is like a moonshot. Why not just doing what is the feasible one first?
First of all, because this is the initial study, actually there hasn't been gene panels that have established for this kind of study to target only why the tumor transformed into malignancy. So that's why we want to whole exome sequence our patients, because maybe we will find some novel mutated genes, and also maybe in the future we can use this data to reanalyze or to find something interesting that can be used to maybe another study with different storyline.
Yeah. I was expecting actually, rather than doing the primary tumors, you guys should consider actually looking at because the ascites you can actually collect. You can actually centrifuge and you get the sediments. Did you even save those from the same patients and run it the exome sequencing?
Actually, yes, we will be conducting that ascites extraction, but it will be our next plan. Yeah.
Right, because if you look at the primary ones, I really worry that you're not going to see differences, because what matters is the one that migrated out from the primary sites. So that's why it will be very more interesting to look at the migrating cells, because they may have additional mutations not found in the primary site. Are you considering that?
Yeah, yes, of course. But yes, we're considering that. And sometimes the acidic sample in our patient hard to get, because we mainly has this primary tumors only sample. So that's why we try to have story here. Maybe we can find something from the primary tumor that gave into the development into malignancy through ascites or something like that.
This choice of 3 non-ascites and 5 ascites, do you think should be sufficient enough samples to make conclusion, or do you think you need more samples? If it's more, how much more do you think to make a conclusion of differential mutation profiles?
Okay, we can see here that it's not sufficient sample, of course, but this is the initial study, and this activity is quite very new, very novel. So this is like a new story. That's why we will validate our cohort to the TCGA cohort, which consists more than 200 samples of established tumor normal samples. So we will validate our finding with that cohort.
Yeah, and also in the poster, I think, because you also collect the normal within quote from the same patients. How are we going to use this information from the so-called normal sites as being a control?
So our normal sample will be used to filter to get only somatic mutation, because during cancer malignancy, often somatic mutation has a more meaningful impact, or has more things to say to interpret what's going on during malignancy.
But how do you know these differences actually causing the disease, actually the driving of having this ascites?
So, okay, so during our study, we will have like our gene of interest that may have meaningful effect on.
Okay, thank you. So, okay, thank you very much for the first round, and your turn, Pa. We'll have the slide up. Okay, that's it.
Wait,
You ready?
yes,
please.
Bismillahirrahmanirrahim. So allow us to present our work in detection of DNA methylation from swabs using next-gen sequencing to study stunting. So in Indonesia, the prevalence of stunting nationwide is about 20%, except in Pandeglang is about 30%. So the major mechanism of stunting is due to chronic malnutrition, and the reason being when you have a less number of nutrient, then you have less numbers of methyl donor, and this methyl donor is required for epigenetic programming to put the methyl modificators in the genome of the baby. So when the baby actually growing up, the pattern of the programming should be seen. And many studies have used blood, and they show difference of methylation when you use blood DNA. But in our study, instead of using blood, we're going to look at the buccal DNA. Since buccal DNA number of DNA is kind of limited, so we're going to use NGS nanopore because they can detect methylation in its native form without having to do chemical conversion. So next is our design method. So we actually recruited about 231 children, 30% of them are stunted, and we collect all the DNA and the 12 highest DNA concentration, consider 6 normal and 6 stunted children. And then we aggregate the data into stunting and without stunting, and we look at a certain loci of differential methylated region using this NGS nanopore. And the data looks like, next slide, if you look at the red arrows, there are areas that have a difference in the stunting and normal, and in the loci of IGF-H9, which is encoding this growth hormone. So if you see that it seems to be consistent with the idea that when you have stunted, you have less numbers of nutrients, so then less number of methylation in this region. We got also the similar pattern in other genes, and this has been published in previously, but using blood. So hopefully, technical approach that we use using nanopore will actually help for the further studies recruiting more children's DNA.
Okay, time's up.
Okay. Thank you for the presentation. And are you ready with question?
Okay, question can start now.
Okay, thank you for the nice presentation. Your objective is, if I'm not wrong, is to justify whether buccal has the same qualified DNA requirement instead of blood. But from your result, you only get 12 from 230. So is that all right if we conclude that the buccal swab is not sufficient or not right to do this kind of methylation study? Thank you.
Right, thank you. So if the data showed that there is no differences in the normal and stunted DMR, especially in loci that has been shown previously, they've been associated with stunting, then yes, that will be useless. This use of nanopore will be useless. But apparently we do see some differences. If you notice in this normal and stunted ones, and this is an aggregated one. So this graph actually represented of a collect together. So the normal legend actually is 6 normal children, and stunting means 6 stunted children. And there seems to be a differences in the methylation. However, we are not going to stop here. We have to do some quantitative analysis, how much actually for each one, because we have to also stratify it according to age and also according to sex. So right now we are still doing some analysis, but this preliminary data show at least this NGS is actually promising that we probably can do further analysis.
Okay, what I want to highlight is the DNA requirement is not sufficient. Is that during the DNA extraction, you cannot get the sufficient amount of DNA to go to the machine, or no? because it's really important to standardize.
Generally, for the traditional classical NGS, they need at least about 500 nanogram until about 1 microgram. In our children, roughly we have about between 100 but 400 nanogram. So we start with the 400 nanogram, with children with has 400 nanogram first. But those children with has less than 400, we can retake the samples if it's necessary, because we know where they live, because this in Padeglang and GRC has a very good connection with them. But we also can think of some other method maybe to pull it together, because we can, using library coding, we can tag them, so at least the total number of analysis should be sufficient.
Okay, so what we have known that actually DNA methylation is kind of tissue specific, right? And then how can you be sure you are not missing like the important or the most essential information, because the blood itself is have many controversial things
Right, you're right. Yes, epigenetic is definitely has something to do with the differentiation, right? But we think that there's published result, and we are very happy because somebody has done the correlation, epigenetic correlation between DNA from the neurons, from the brain, from also the fibroblasts, from the buccal, and from the blood. And the correlation is quite high, it's higher than about 90%. So it's not that only about 40%, but 90% is pretty good enough. But this is going to be validated by looking at different loci, because some other publication have another different candidates that we can look at, and that could be something useful, because getting buccal definitely is easier than getting from blood.
I think what hasn't been highlighted in the presentation is that for the Oxford nanopore, and then you do all your analysis and everything. Has it been benchmarked? Has it been validated by? Has this pipeline, data analysis pipeline, been benchmarked, been validated? And why would you use aggregated IGV instead of like say examining all the samples individually? Because I think for each of the samples, they may deliver different variations. Yeah.
Right. So we collaborate with a lab in UK, in Nottingham University. But Ninin has been very involved in nanopore, developing nanopore for COVID. So she has actually done this quite a lot. And also the way we benchmark it, the method is called adaptive sampling. So what adaptive sampling does is actually focus on areas that we want to sequence, not the entire genome, but only the CPG islands. And we benchmark it with the the Infinium Illumina. So in Infinium Illumina is microarray based. So they look at 30,000 sites of the genome. So we make sure that what Badinin did, he makes sure that all these 30,000 sites of CPG islands actually exist one being read by nanopore. So this is a good thing about adaptive sampling. We can choose which area we want, because if we don't choose this area, then we cannot benchmark with these genes. So alhamdulillah, this gene has been found, and looks like we have a similar trend, but we're still working on the quantitative analysis. Hopefully, maybe within next week, we're going to see the actual numbers, because this one is easy to visualize. At least we see some major areas of differences, but you're right. Definitely, we're going to do some percentage analysis.
Okay. Anything else? Because my question about aggregation is your.
Okay, this is an interesting research. What's your next plan? Maybe you can tell the limitation of this study, and then what your next plan to.
Yes, so people have thought that.
Time's up.
Okay. Jadi Anda pakai primary tumor ya? Itu pertanyaannya bagus, untuk apa primary tumornya bisa dioptimalkan untuk apa lagi gitu kan? Berarti kan bisa dispesifikkan lagi nggak kalau pertanyaan Bapak tadi? Iya. Dioptimalkan kan saya kenal Dr. Vero. Oke. Dia patolog di dunia. Jadi memang
dari dulu saya juga sempat mikir sih, kan asites itu kan juga banyak sel, bahkan untuk ini untuk sel lain itu kan dari asites.
Iya.
Selnya itu agresif banget di asites itu, gitu. Makanya saya pikir nanti mungkin coba dipertimbangkan ya sama coba dilihat dari asitesnya gitu. Karena itu yang membuat kuncinya.
Iya, oke. We have the final
mungkin
score.
diferensiasi terlalu banyak juga ya. Oke. Mana? Belum? Oke. Oke, congratulations.
Ini Pak, sebenarnya kita tuh sudah mau mencoba, tapi
Bapak ingin nggak?
Iya benar.
Sama lagi. Siap. Oke, congratulations.
Sama-sama satu.
Jadi asites kan selnya banyak banget. Kalau bisa ketemu jinnya apa gitu.
Kita di mana tuh? Room 1 tuh mana sih?
Iya, ekstraksi dari asitesnya. Oh. Oh, Anda ngambil dari biopnya ya? Bukan langsung dari pasien asites atau dari pasti bisa banyak. Banyak. Iya, bisa begitu itu. Jadi pasien dengan cancer yang ada asitesnya bisa boleh trakonnya. Ya, jadi bagus. Tinggal di defining aja.
Apa ya?
Anda di biarkan mulai di Oke, betul. Ya, tapi kalau bisa dari asites dari pasiennya, Pak. Soalnya ini juga bagus tuh. Mati deh. Ini Pak Ahmad sama. Ya, mari sama-sama sukses. Ya. Ada bau kopi.
Iya, Pak.
Udah ngantuk. Kayaknya menarik nih. Next ya, terakhir untuk sebelum break ya. Oh, belum dong. Tapi
Saya udah.
Tapi kan tadi
Makanya kita keluar.
Ya udah break dulu deh sebelum ada. Oh, gitu.
Oh, udah.
Kampus dah. Kita keluar aja. 10 menit.
Itu agen
Oh, ikut. Oke. Cerita aja. Ikut.
Nah,
Oh, iya. Si juga ya. Yang di Bulu itu. Selesai pakai pada
oh di kantor ya?
Di kantor.
D13 and D16. D13, you're over here. Your match is ready. D13 and D16. Okay, D13 and D16.
One, two, three, check. Cek. Suara 3 Pak. Cek 1.
Udah. Oke. Ulang lagi dulu. Pakai ini ya, biar masuk itu dia. Siap. Kita satu lagi sebelum break.
Udah mulai break ya Pak? Breaknya tinggal 5 menit lagi.
Oh gitu. Sekarang ini break ya?
Iya.
Berapa sesi lagi?
Habis ini lanjut ke match 28, kemudian match 31. Ini lanjut sampai
Berarti 2.
sholat di 3 lagi sebelum sholat.
3 lagi sebelum sholat.
Berarti ya 1, 2, 3 baru kita break sholat.
Mbak Yudis,
Iya,
ini match berapa?
28.
Baru mau 28.
28, 31 dan
Oh ya udah berarti mau
28, 31, 34 baru break sholat.
28.
31, 34.
Oh karena udah break baru.
Iya.
Kan ini 3 ruangan masuk gitu gimana? Semuanya masuk gitu.
Jadi kita ada 3
channel
view gitu. Iya. D30 and D51. We will start the match.
Dia bisa makan apa nggak, bisa bicara apa nggak. Cepat gitu.
D30.
Yes.
D30, and this one is 51. Okay. Welcome back. Okay. Do you wanna still introduce yourself before we start? Please.
Introduce.
Yeah.
Hello, we are from team D51. My name is Christy. I'm a pediatric neurologist from UGM Academic Hospital. And this is my colleague, Dr. Aulia. She is a pathologist from Dr. Sarjito Hospital, Yogyakarta. Thank you.
Okay, good afternoon everyone. We are from D30 team. My name is Indira Prakoso, and this is my teammate, Fahru Rizal. We are from InBio Indonesia.
Okay. Do you have any question about the role on how we want to go? You are okay, right? So this is not the first time. So I'm Ahmad Hidayat. I'll be the referee for this session. There's a Pak Iqbal. Where is Pak Iqbal? Yeah. Okay. We'll be the judge, and Bu Ati also the judge. And we have the team to do the timer stuff and admin stuff. Okay. So we want to have a toast so that you can choose which going to be the one to present or to defend. The winner of the toast would be the one to choose. Okay. Which one do you want to choose?
Number.
Number. This one, yeah.
Yes.
And you are this one, yeah.
Yes.
Okay. Is a number, right? Okay. So you choose. Present or defend?
Defend.
Defend. Okay. You present first. You present first. D30.
Yeah, D30. Okay, ready? Okay. Can I start? Can I start yet?
Ready? Timer? Okay. Yes, please.
Okay, good afternoon everyone. So here is our research about bioinformatic analysis of microRNA mediated regulation and their potential as therapeutic targets for lung cancer. Next. So based on global cancer data, it has known that lung cancer has become the most lethal cancer. Previous studies show that current cancer treatments such as chemotherapy and radiation still face high mortality rate. Therefore, we need innovation of treatment for lung cancer. Previous study has also shown that microRNA can act as tumor suppressor. The organoprotein can help the microRNA to degrade the mRNA. So this research aims to identify the most potential microRNA as the novel therapeutic approach for lung cancer in stage 1 until 4. This research utilized the microRNA data in TCGA database and analyzed it with bioinformatic approach. Next. So here's the heat map and ANOVA analysis show significantly different expression level of microRNA between the normal and cancer sample. The volcano plot split the microRNA based on the expression level, the expression profile. And the table here shows the list of the most potential tumor suppressor microRNA in stage 1 until 4 and its target gene. Next. So the selected microRNA have been evaluated with RLC analysis. Meanwhile, the target genes shown to be involved in the pathway and biological process of cancer. And the molecular docking show highly binding affinity between the microRNA mRNA duplex and organoid protein. And this is the visualization of the docking. And the conclusion from our research is these 4 microRNA have potential to be developed as therapeutic treatment for lung cancer in stage 1 until 4. But further validation still needed with in vitro and in vivo. I think that's all from us. Thank you.
Time's up. Time's up. Okay. Are you ready with the question? Okay. Timer.
Okay. Thank you for your presentation. Okay. In your poster, you say the data is from normal patient and normal patient of lung adenocarcinoma. What do you mean? Because if there is a lung adenocarcinoma, it's not in a normal patient. What do you think about that?
Actually, we use the data from normal patient and the lung cancer patient. I don't know if in the poster we might be typo or something, but yeah, actually,
yes, I want to confirm.
Yeah, actually, the data that we use is the normal patient and the lung cancer patient. Only that.
What the sample do you? It's from the blood paraffin or blood or something like that.
Okay, this sample from the database. The database informed that the sample this from the blood and the solid tissue. This mixed blood and solid tissue and the adenocarcinoma cancers. This is a mix.
So that is from the normal patients and the lung cancer patients, and
Yes.
you compare between them.
And when we work for the gain expressions, we must to consider the normal sample to compare with the cancer.
What is the genetic background of your database? Is it Caucasians or also Indonesians?
This is the TCG database is global database.
And it concludes of the race and ethnicity from white, Asian, black, yeah. So the diverse people in that database.
Also Southeast Asians, Malay.
Yeah, all of the all of the race and ethnicity in the global.
And you say in conclusion, you correlation between mRNA and protein association, and how do you examine the protein in this study?
Can you go back one slide? Yeah. So here we use target scan prediction. It is a web server, and based on the total contact score in the mouse right, the lowest of the total contact score means that the target gene is the best match of the microRNA potential. So we use target scan prediction of that.
So in clinic, you will check the target gene first, and then you will choose which microRNA that match to the target gene of the patient.
Yes.
So what is your next study to confirm which one is the great Mirna for the lung cancer?
Yeah, actually there is one step that we have not done in yet. It is the molecular dynamic to validate the molecular docking result. And then for the wet lab?
And maybe the wet labs, we can use the in vitro analysis. We can inhibit maybe the stem cell, the specific cancer. Maybe I forget about this cell, and we can analyze about the profiling transcript topic from the cell lines. And maybe next we also use the liposome encapsulated microRNA, because microRNA, we know the microRNA have the instability structure that have directly degraded by the several enzyme, and we consider to meet the liposomes nanosolubility.
Yes. Do you know in Indonesia now we can examine the DNA target from like therapy for EGFR, like that. What is different with your study? Because DNA is more stable compared to Mirna.
Yeah, so actually the microRNA can targeting more than one gene, multi-target. Is that answering your question?
Yes, but is how about the feasibility in Indonesia? It's more cheaper than DNA target.
Yeah, actually, we propose an innovative treatment. So and we have not analyze about the economical feasibility about that, but this can be used to increase the to decrease the mortality rate of lung cancer patient. I think that because can you go back to the one slide? Because based on the reference here, the current treatment such as chemotherapy and radiation still face high mortality rate. So I think the innovative treatment of this is still needed.
Yeah, but the targeted therapy is already in clinical nowadays in Indonesia.
Yes. Yes, targeted therapy like EGFR and immunotherapy already used in clinical practice in Indonesia.
Time's up? Yes. Okay. Okay, thank you. And then we got a next turn is you be the presenter.
Thank you. Our study is about exome sequencing for personalized treatment in DEE patients, epilepsy patients, transforming national clinical practice guidelines in Indonesia. Next slide. So this is the most severe group of epilepsy. It causes severe morbidities, severe seizures, drug resistance, and cognitive behavioral impairments, and also early death. And you can see there's a lot of genes responsible for DEE, and if we can know the gene responsible for underlying mechanism and inheritance, we can transform the clinical diagnosis and also management. Next slide. Current guideline using the therapy based on the type of seizures, which might be contradictions of underlying mechanisms. For instance, for focal seizure, we always use carbamazepine, which is a natrium channel blocker. But if we test the DNA is SCN1A, which is also a natrium channel blocker. If we give the CBC, the seizure will become worse. The study aims to look at the genetic profile of DEE patients, directing patients' therapy and management in Indonesia. Next slide. So the DEE patients, we phenotype and do the whole exome sequencing to know the gene responsible for our patients, and we do the personalized treatment based on the gene we found. Next slide. We identified the genetic etiology in 47% patients, and the most common genetic etiology was SCN1A. That's why our patient has been given the wrong drugs. Management changes was conducted in 9 patients, 7 drug changes, and 2 avoidance of brain surgery. Next slide. So our study illustrates that whole exome sequencing in clinical setting in Indonesia is feasible, has high diagnostic and clinical utility, and modify the treatment for DEE patients in Indonesia. Thank you very much.
You still have 20 seconds. Do you want to add more?
Thank you.
Thank you.
Okay, thank you for the presentation. The question, are you ready?
Yes.
Okay,
Timer?
let's start now.
Okay.
Okay. How much the sample that you use in here? We read from your poster that you only use 50 patient, and we think that your sample is too low to represent all of the Indonesia, because you refer in the title that you recommend it for to use VES in Indonesia.
Yeah, thank you for your questions. So this is a rare disease. It happens in 1 every 1,000 persons. So it is rare. And we collect all the patients come to our center, tertiary referral center for 2 years. And compared to international studies in Australia, the whole national Australia studies, they use 100 patients. So I think our number of sample is quite reasonable.
But how do you assure that your sample can represent all of the Indonesia?
Yeah, it might not represent all of the Indonesia, but as a tertiary center in Central Java, we receive from the west part of Indonesia and also from the east part of Indonesia. We have to, yes, you're right, we have to conduct much more multi-center studies to make our data more reliable. Thank you.
And then can you show the diversity of your sample, the distribution, like the demographic and then the race, ethnicity in your sample? Can you show that?
Yeah, we actually collected the data, but we don't show you here. And yeah, maybe on later we can show you the data of race and ethnicity.
All right. So about the, you said before that this method is quite expensive, isn't it? Yeah. So but in this conclusion, you said that the WES in clinical setting in Indonesia is feasible. So is it efficient for the economical condition in Indonesia?
Yeah, thank you. So the price of WES is 5 million. So it is actually quite high to Indonesian economy, right? But from the economic health perspective, if you can use the 5 million to stop the seizures, you can avoid the 50 to 100 million rupiahs for brain surgery, and you can stop the disease, the morbidities, to have yearly of seizures and complications, pneumonia, PICU treatment. Yeah, and that's actually a price of a life. So I don't think that's expensive.
But we have research before that there is already research that use PCR method to diagnose the development and the
No. Maybe you can only one. For instance, I don't know. I don't think SCN. Yeah, maybe only for one gene, SCN1A. But as you can see in the previous, maybe you can show the first slide, the second slide. The phenotype is once more. Yeah, the phenotype is very look alike. You cannot different between the patients, and you cannot differentiate between the MRI. They are all look alike, and there are more than 400 genes responsible for this type of disease. It is very very difficult to use one gene to one gene to one gene if we do the PCR. But whole exome sequencing, you can find any genes.
Yeah, but in the research paper that we read before, it used more than one biomarker. So I think it's feasible to use PCR.
No. Oh, could you elaborate more about the paper, because I'm in this realm. So yeah. I use PCR for DMD because I can see I know the suspicion of one disease for Duchenne muscular dystrophy. I know this is the Duchenne muscular dystrophy, so I just use PCR. Multiplex. I use the MLPA actually, but I know that is single gene testing. But for this, it is very hard to use PCR for this DEE because so many genes responsible for these patients. Even multiplex PCR is impossible for 400 genes.
Okay then. And then, do you use did clustering of the mutational gene about the surveillance and management treatment?
For the statistics.
To
determine whether mutation of the gene have the impact maybe in the phenotype, and how treatment to treat the patient about the specific mutation.
Yeah, thank you very much. We haven't conducted the statistical analysis data, and we will do in the next study also economical data for utilize this WES in our clinic. But that we find that 9 patients out of 50 patients changed the management and has a good outcome, and also we save like I said two brain surgeries. That's a rough estimation.
Okay, and then in the
time's up.
Okay, thank you. Di Singapura bukannya udah dilakukan screening carbamazepine kan?
Oh, kalau itu buat second Janssen pakai HRA.
Oh, dia buat second Janssen aja.
Iya, dia buat efek sampingnya.
Oh,
iya, bukan buat
efek effectiveness terapi.
Tapi sekarang sih Singapura juga semua. Ini sih orang kita banyak
Yang
di
ini.
sana.
Oke. Berarti ini langkah tapi masih jauh nih untuk HTA ya. Saya ada beberapa step lagi ya.
Iya. Kecuali kalau pakai BPJS nggak mau. Tapi sebenarnya sih untung sih Dok. Coba itu kan kalau pasien kayak gitu dia bakal datang sampai meninggal. Ya kan?
Ya makanya ini kan kita bisa bikin cost analisisnya kan.
Sebenarnya.
Dengan HTA misalnya oke lah dia pakai BPJS, berapa sih dia BPJS yang harus dikeluarkan sampai dia meninggal gitu kan, dibandingkan kalau dia ambil pemeriksaan ini langsung ketemu. Cuman Anda harus bentuk ini menjadi kit testing ya kan, supaya itu arahnya tadi multi center bagus tuh, jadi multi center. Oke, got it. Congratulations. Oke. Apa bikin kit, Anda bikin multi center, datanya lebih banyak, ya kan? Terus tadi yang switching terapinya berhasil. Terus kita bikin HTA-nya, sehingga apa namanya kita bisa compare kan efektivitasnya orang yang nggak tes sama yang tes. Banyak kok yang mau sponsorin.
Gitu. Ini kita lagi nyari GSI. Gimana GSI?
One match before we break for
Bagus banget. Sebenarnya saya nggak punya pertanyaan.
Apa Mirna itu punya potensi besar untuk novel terapi, cuman Anda sulit banget untuk isolationnya. Itu satu. Cuman pertanyaannya untuk masuk ke clinical aspek, nah itu memang challenging.
Makanya kita mainnya di bioinformatik.
Kita nggak tahu bioinformatik.
Kalau strateginya masuk ke clinical guidelines.
Okay, do we have team D49 and D14? Yes. Right now before we break.
Oke.
Kalau mau ke HTA sama dokter AHI.
Oh, iya. Menarik ya.
Kalau di sekolah kita pakai feedback kan.
Oh.
Jadi karena mereka kan ada waktu coaching 1 menit ya. Jadi kayak break gitu.
Itu bukannya mereka punya sensei masing-masing?
Iya, tapi kan ada nih sekarang. Tapi karena itu semua nggak ada kan. Jadi kan mungkin nggak tahu strateginya. Minggir gitu. Jadi kalau kita kasih feedback buat semuanya gitu. Terakhir kita kasih feedback nih. Ini gini. Tapi nggak spesifik itu, cuma kita bilang presentasi nilainya 3 nih. Dari tadi kita belum lihat yang presentasinya brightening gitu misalnya. Jadi memberi kode kayak kalau iya. Itu kata presentasinya di sini. Tadi kita main selesai dulu aja nih ini. Sudah.
Oh, datanya ada. Oke, iya itu betul.
Tim satunya mana? Masih bisa.
Halo. Siap habis itu. Kita harus sholat ya di sini.
Di 14. Baru asar juga kan?
Iya, baru asar.
Mbak sendiri nggak kan? Berdua kan?
Berdua.
siapa yang duluan itu?
Nggak tahu, taruh aja dulu.
Ya udah nanti taruh dulu benar. Iya benar.
Cek untuk penggunaan mikrofonnya tidak lebih jauh daripada 5 cm biar suaranya masuk ke rekaman. Ya, terima kasih. Okay, is everybody ready? Our judges and our team? Okay.
Can we start?
Okay. Can we start now?
Okay, thank you for joining this session, yeah, D14 and D49. Okay, so yeah, I'm Ahmad Hidayat. I'll be the referee for this session. I think we can start first with the introduction. Please.
Hi all, my name is Yeni and I'm from Badan Riset dan Inovasi Nasional.
Hi, I'm Fikriya Hanum Humaira and I came from the same institution.
I'm Lina Herliana from BRIN as well.
And I'm Hayrunisa from BRIN as well, so all of us are from BRIN.
Biotech?
Yeah,
Applied microbiology.
Genetic engineering.
Genetic engineering. Okay. So we want to have this session. Do you have any question about the role? Everything is clear? Yeah. Okay. And then we have our judges, Pak Iqbal and Bu Ati, and also the technical team. So we want to have a toast to decide who's going to be the first, yeah, to present or to defend. Okay, which one do you want to choose?
Number.
Number is one, yeah?
No, number is.
Oh, this one, okay. So you this one, yeah?
Yeah.
Okay.
Face.
Face.
Number.
Number. So you choose number, yeah?
Yeah.
Okay. Oh. You want to present or do you want to present or defend first?
We present.
Present? Okay, please. D49 will present first. Do we have a slide? Okay, we have a slide. Timer? Are you ready?
Yes, sir.
Okay.
Okay, good day, my lovely audience. We want to tell you about our story, Journey to the East. Next, please. Okay, we went to Sulawesi, the eastern part of Indonesia, to study the most expensive wood in the world. Now, maybe some of you are asking, do we really have that? Or why do you have to go that far, thousands of kilometers, to study that?
Well, yes, we have ebony, an endemic to Sulawesi. Ebony is one of the most expensive wood in around the world because of its strength and durability. However, based on IUCN, ebony is listed as a vulnerable species and one of them because of the plant disease.
Then we came up with this idea. If we can understand, the previous one. If we can understand the microbial diversity, in this case bacteria, from the asymptomatic and symptomatic leaves of the ebony, we can formulate the natural biocontrol against the pathogen causing the disease.
Next. At first, we thought that the symptomatic leaves will have more bacterial pathogen. However, we found a very interesting data where the symptomatic leaves actually have more potential bacterial group. So as you can see in the genus level, Spingomonas and Bacillus are actually abundant in symptomatic leaves, and those bacteria are highly reported for their antipathogen activity. So how can it be happen? So this is maybe one of the strategy of the plant to survive from the pathogen or stress condition by recruiting the bacteria or beneficial bacteria to help them combat with the situation. So the next is we try to isolate those potential bacteria and then formulating as a biocontrol agent for the survival of the ebony. So we are team D49.
Thank you, and we are open for discussion.
Okay, thank you for the presentation, D49. So we now have D14 to ask the question. Are you ready with the question? Yeah. The timer? Okay, yeah.
I think you have a problem since the beginning of your sampling. Why? This is the reason. If you look at your sample, you don't have any replicate. So it's only 4 in each group. So how you define this thing? Why you didn't do any replicate?
Okay, thank you for your question. Yes, you're right that we don't have any replication. So our idea is to composite all the sample that we found in the natural forest. For example, we sampling in the forest sampling site, and then we combine the symptomatic leaf sample and then asymptomatic sample into one sample, and then we send it into the genomic or surface of the metagenomic analysis. But actually, this is a preliminary data, so we don't try to make a representative of this result to all the population of the ebony. So the end result is we want to know what is the bacteria and try to isolate them and then formulate it into something like that.
So do you think it will be different if we use the replicate compared to only use one?
It may be, but once again, if we use the replication, it is also we need to composite the sample, right? And then with the sample, we do some replication, and then we find the result of that, and then maybe we found almost similar result for the one that we found here.
I think it's the bad thing to actually pulling the sample because you lose the diversity between genotype. I mean, between the biological replicate. The other thing, there is no statistic in here. So this result is only description. If you look at in here, there is no bar. I mean, there is no standard deviation. There is no what this is significant or not significant. Whether or not your conclusion is based on a statistic. I mean, the backup from the statistic.
Yes. Our study design is a descriptive study, so that we don't have any replication. And as I said, this is a preliminary study for us, so we know what kind of bacteria here, and then is there any potential bacteria that we can isolate from the sample in here, and then after that we isolate, characterize their beneficial function in the plant, and then formulate the biocontrol agent for that plant.
The other thing. Because this is symptom asymptomatic and is already in leaf, it means that already passed from the, it's possibly from the root, from the hydrosphere and into the top. If you look at the end, do you think you can prevent if there's, because you don't know the plant disease, have you tried to screen first from the, hydrosphere until the leaf?
No, we only do that in the leaves. Why? Because the symptomatic and asymptomatic symptoms is actually can be observed in the leaf. So we don't do any sampling in the root or in the soil or etc. So that we choose first of all the preliminary study in the leaf, and maybe in the next future plan we can do that as well.
Do you think you can publish this? Yeah, is it novelty and insignificant?
Yeah, sure. Why? Because this is the first, I think this is the first report for the metagenomic analysis data for the ebony, especially Diospyros celebica bog, endemic to Sulawesi.
I want to add something. We work with forestry commodity. So forestry commodity ebony actually can be harvested in 40 years old. So it is quite impossible, maybe possible, but it is hard work to analyze in the several stages, such as in the vegetative and generative. So
I don't think. Sorry, my question is: you can still take the root. I mean the hydrosphere. So you can prevent at the beginning of the infection rather than direct already at the end of the study. I mean, if there is a data, your data, for example, comparing up and at the beginning, it will be more effective for this prevention.
Okay, first of all, ebony also live in the karst area and the stone area. It is also very difficult to find the soil or to make a representative soil for this. So at first, our preliminary study, we try this leaf because it is clear between the symptomatic and asymptomatic observation. So it is very easy to see, not like soil or root or maybe the stem.
Yeah, that's the reason why you need to do the metagenomic, right? You take the sample that from infected plant and in not infected plant, not only one plant, and then you look at only one spot. So if you want to know that whether or not this microbiome is different, you need to compare between plant in the area that infected and not infected.
Actually, this is not the end of our result. So I think that this is only the how we can choose the dominant bacteria, and how later on we select the bacteria itself for the formulating of the biocontrol agent. So the end is we want to formulate the biocontrol agent, isolate of this bacteria, and apply that for the plant, ebony plant, for the conservation efforts.
Since the cause of the disease haven't been identified, how can you be so sure that the bacteria is biocontrol agent, but not instead of the cause of the disease, because it hasn't been identified?
First of all, is like we already also have metagenomic analysis of the fungal, and we found that this is the fungal. This fungal is actually have the pathogen that is similar symptoms to the the symptoms in the leaves in the natural. But so this is the
So
reason
that
that
fungal.
Yes, fungal. Yeah.
But you said it hasn't been identified. Why don't you just state it?
Thank you. Okay, then we will have the 14 to present. The slide is ready. Are you ready?
Yes.
Timer?
I think.
Okay, start.
Okay, good afternoon, everyone. So our study is about Plantago ovata. Now, Plantago ovata contains mucilage which has significant economical value, but unfortunately, the mucilage content is only like 25% of the whole total seed weight. So we aim to increase the mucilage content by identifying the genes that are involved in mucilage biosynthesis in Plantago. Sorry. So we do that by comparing the transcriptomic profiles between the wild type and the mutant ray. Now, the mutant has higher mucilage content than the wild type. So total RNA was extracted from two different types of seed tissues. The first one is the integument, INT, and the second one is the mucilate secretory cells, or the MSC. Now, we took the samples at 2 time points, 2 and 4 days post-anthesis. And for the method, we use the RNA sequencing analysis. Our hypothesis is that the transcriptome profile in both tissues would be different between the mutant and the control. Now, for the result, we got around 25,000 genes expressed in our samples, in all of our samples. And by further analysis, we find out 18 genes in INT and 8 genes in the MSC were expressed higher in the mutant than in the wild type. And so those genes are related to the xylem biosynthesis, which is the main component of the mucilage. So I think they're important. So we did t-test on the 22 genes, and we end up getting 2 genes with significantly higher expression in mutant than in the wild type. Therefore, we prove our hypothesis. Thank you.
Thank you, D14, and we will have D49 to have a question. Are you ready? Yes. Yeah, then time? Okay, we could start now. Yeah.
Okay, I see that you made a mutant of Plantago ovata using the gamma irradiation. First of all, regarding that, do you have any ethic clearance since you are working with the genetically modified organism and it is still controversial in the world?
Thank you very much. If you know that this is mutation, is not CRISPR. This is it's conventional, and then a lot of we use the gamma radiation, so it's not something that you need to do ethical clearance for the gamma radiation. But we do have in the local at the university. Thank you very much.
So I see that you do the transcriptomic analysis of your plant, and so actually, how much sample that do you use for this transcriptomic analysis, and how do you calculate the sample size?
So we use 32 samples, and at least in each category we have 4 replicates. Yeah.
From where do you obtain the samples? Because I see that the plants is specifically location grown. So is it generalizable for all population of Plantago?
Okay, so we selected another project. Before this, we selected 2 genotypes, so the wild type and the high production of mucilage. And once that we grow in the condition inside the growth room, So it's it's not from the outside, because this is already screening until 5th generation.
So before you do the transcription analysis, and what criteria do you use for the screen the mutant array? So so what is the eligibility criteria of your study?
So this produce a really high mucilage and is significantly different between the wild type and the mutant. And the other thing, we don't want any other effects, so we screen the phenotype from the plant itself, the growth should be similar, and then from the seed morphology should be similar, and the component should be similar. The difference is only the production, the yield.
However, I found that the one of the gene that is actually expressed more is about the ethylene production, ethylene transporter something. So I think this is one of the, like if it's too much production, it will inhibit the growth of your plant itself. So this mutation is actually also disturb another metabolism of the plant, for example, like growth, chlorophyll or something.
So the main finding is actually we are looking in the mucilage, the biosynthesis of xylose transferase. So it's a xylene. So it's a composition of the mucilage is xylene. So we are looking more intensively to the xylene genes.
But if it's good for that, but not good for the growth of the plant, so it is also influence the production of the seed as well, right?
Because we already done the morphology screening, so we only look at the genes that actually, we already any possible bias we already filter.
May I add? So she has observed the seed development, and it doesn't affect the mutation doesn't affect the seed development.
Okay, regarding that, how can you be so sure? Did you analyze the quantitative real-time PCR? Because RT-PCR is actually can quantify the gene expression.
But RT-PCR only 1 or 2 genes, right? So at the beginning we need to do RNA-seq. The best thing, why is RNA-seq better than the qPCR? Yes, qPCR will done at the end, but at the beginning you want to look at the whole thing, any possible expression of the gene. It's not directly 1 or 2, because model species is so different from this plant what we are studying. They are having a pectin mucilage, and this one is heteroxylene, which is
yeah, but I think it is still important to see the quantification, because we need the targeted genes to be more abundant than the ethylene gene.
If you can see from the standard deviation, they're pretty narrow. So I believe if we were doing the qPCR, it would have the same similar result with this one.
So also how you handle your sample before the transcriptomic analysis, because RNA is very sensitive and it can make your easily degraded, and also it can maybe not validate your data.
Okay, so what happened is I spent like 1 year to optimizing this thing. So all the same is randomized, and then all is a really quick procedure doing cryosectioning. So all already in the cold condition. So we know that we prevent the degradation of RNA, and then before we go to the sequencing, the all need to be the same amount of the RNA, and then we get the whole thing after the analysis, and then we do the filtering as well using the p value less than 0.05. Then we got that expression.
So after you collecting the sample, do you preserve it in the freezer for 1 period, I think?
No, so RNA you should directly, once you get it, you put it in the liquid nitrogen, and then directly you put it in -80 until you proceed.
Okay.
Enak ya dengerinnya.
Soalnya mirip.
Mirip ya. Okay. I think it's a very entertaining discussion. Okay, the judges is collecting the result. Tapi kan metodenya beda kan, yang Anda lakukan kan beda kan, ada metabolomik untuk tahu plant diseasesnya, Nah, apakah Anda yakin bakterinya itu? Oke. Gimana Anda yakin itu adalah plant disease?
Sebenarnya sih kalau di plant disease itu kita belum yakin apa. Tapi
Nah
kita
itu,
sudah
mestinya itu yang Anda tanya.
Sebenarnya kita udah isolasi fungal patogennya dan
Ah.
itu merupakan di referensi itu merupakan fungal patogen bagi wood plant wood yang lainnya.
Oke,
Tapi
belum
salah
tentu di
satu
ebony.
konfirmasinya itu adalah postulate cause. Jadi kita harus aplikasikan fungal itu juga ke si eboninya untuk melihat simpansinya.
Now we can start for break for 15 minutes. We will break here at 4:10. Thank you.
Jadi Anda bisa jadi dokter. Ebon.
Ini minum itu. 15. Terima kasih.
Nggak lebih.
Jangan Pak, ntar molor.
Oke.
Ya, satu kali lagi. Ya. Didampingi kan? Berapa lagi kita?
Yang sudah ada namanya satu dok. Biasanya kita akan ada match.
Diadu yang menang-menang?
Betul.
Belum ada?
Sudah ada namanya match 34. Nanti kita tunggu match 35, 38, 39, 40 terakhir di kita.
Berarti kita nunggu jadi?
Untuk yang setelah ini langsung ada, tapi
setelah itu
kayaknya mau dipecah ke ruangan lain jadinya ya, karena ruangan lain sudah selesai, tutup.
Oh.
Tutup warung.
Tapi nanti ada beberapa match yang memang harus nunggu match sebelumnya gitu. Yang harus berurutan memang. Jadi nggak bisa bersama-sama.
Laptop yang buat regis di sana ya?
Di mana? Di ruangan sana.
Kayu di atas meja. Kayak.
Itu kan laptop yang
kayak ini dan sini ke arena 3 jadi tidak kabur kan ini udah selesai kan? Ya mudah ya 34 kita 34 absen 34 habis 3 itu sampai sini berarti nang sampai sini aja aku tidak itu nomor 25 naik 34 dia 34 dan seterusnya ya gue pernah ngasih minta
nanti yang
Tahu
dari
nggak?
sini kayaknya dok, jadi kayaknya nanti yang dari sini kita akan pindah ke arena 3.
Digabung.
Sudah kosong mungkin, harusnya dia udah closing kan.
Ah.
Jadi makanya yang dari sini kita pindah karena lebih stabil di sana.
Oh, koneksinya.
Iya.
Oke. Tempatnya aja kan ya. Oke. Pindah ke arena 3.
Di arena 3 tapi.
Tahu iya kan? Saya juga nggak tahu. Mungkin yang di ruangan ini udah gitu.
dilakani. Oh, mikir kan? Lu wis nyate tebus. Ngapain? Ini berarti teko bar newang sebelah. Kabeh. Lah iya iki ngene sak kanggo. Yo wis ah. Beneran ra kerja kok nop to iki bingung. Besar wono. Ruang telu. Bone anu Iwan. Sarwane panel kono apa nyebutne?
Terus bisa 1.000 apa 300.000.
Bukan dari jeret. Ini Mas. Ngomongnya pindah sebelah kabeh. Pindah sebelah. Jadi serah kanggo. Oh, no.
Ya wes bener lah.
Iya.
Orang mau istirahat sebenarnya ini.
Mau dari istirahat sih saat sekalian berarti.
Itu mana? Oke. Dari sebelah ruangan itu masih stabil mas. Dari baik sini yang mau. Tapi dalam kopi atas itu yang api. Sudah loh.
Kok apa mikirnya sama internetnya? Internet tadi sempat ini sih.
Itu mana? toko ning itu barang yang sakitnya malah aku ora duga
makane
mak tidak itu pikir atau benar sakit sasi ya kuwi. Ya iku ngurusi mat. Wis besar awakan kuwi nomor sari wedding wis 11. Ya caca ngene iki gemel sing Andi ya. Zaman kamu sari kayak go PT karena melu campur sarian ya ambil lah ambil 10.000 sudah neng PT jadi kan Mas Agus lah
oh Mas Agus
kan Masku kan
Mas Kandung
di bawah gitu campur sari
lah ini mau campur sari wong gantung biar memang
cuman aku biar suwe kerja neng studio soalnya jarang neng lapangan motet iya apa iya ngedit iya biar neng studio Cenongan
oh M
ya Cenongan pindah neng Manggarai Mbak kita nggak pindah
Jelas. Apa biar Bapak istirahat?
Alhamdulillah Cenongan enek 5 tahun enek anak betul iya Joni oleh
Joni bulannya Bekasi
Joni Bekasi we kopet Cenongan ya Mas
Iya wale.
Jenongan buka nang Manggarai, kowe buka nang Manggarai ngajak aku. Aku acara tahun Juni bro, 13 acara gak dibayar.
Sampai mati.
Iya. Sampai pengeran. Makanya aku metu ke Manggarai itu aku buru-buru terluas acara lagi gak dibayar. Jaluk aku kok Jenongan kan pilihannya Manggarai itu. Bose sing Jenongan sampai nangis-nangis sih. Kamu jangan
dan rejeki kan wis udah kebanyakan.
Iya. Kamu jangan ikut Joni, kamu mau gaji berapa di sini saya kasih. Enci karo engko sampai nangis-nangis. Nyidang aku wong telu ini. Aku rasa aku gak melu Joni. Mulai kampung salahku kuwi.
Berarti yang kadang bisa ngatan karo anu ya Mas.
Coba.
Sebenarnya jane ya wes.
Hah?
Wes ngomong nggot.
Sing wong gumolong wong sakit juga lho. Sing ora rapi apa? Sing tuwek sing biyen tukang sapu apa?
Kebon.
Nah, kebon. Wong daerah sakit juga kan?
Iya, suka kebon. Anda sakit.
Wong nggot iki Mas.
Oji.
Catur.
Jadi ini wis ya.
Wis saiki.
Untuk rondo apa?
Ora tau sapa pusat biasa. Semenjak corona jelas ora tau dolan Jakarta.
Aku selalu ini melucu nih asu. Kebon sana. Karena aku wis apik nang Jenongan kuwi wis tak ngedit ae lah. Wis enak malah.
Sing malah dari operator lah.
Suro ke sak balaku.
Angkatan.
Oh. Suro saiki moto deh.
Tapi yang ada iki cek.
Wis belah ora tau.
Ya tau wong sing keseringe tak ono.
Saat turun meneh aku isin. Dene pengen yo enak sing tak biyen lucunya gak gajian mak enak.
Terus modelnya?
Jadi ya dia.
Oh, berarti kecil lah sih bayar.
Giliran aku wis out metu, aku tengok nang kono. Saiki bulan nenek celeng. Kok jangan kan aku jagane kan yo kok titipku iki.
Iya iya kan.
Gue lah nengeki
Ngadong edit.
Kowe aku ora juga wong amate. Iki wong istana wong iku.
Soalnya kan apa? Ya wong intine wong sapa wae kan nang iku.
Bang aku ngedit lho. Rokok mbok 5 bungkus. Ket 100 wis mesti dino ya. 5 bungkus 3 bungkus sih mesti enak anggere hampir dino anggere ngedit.
Anggere ditongkrongi.
Kowe umi itu di bosmu. Umi itu di bosmu.
Oh maaf, boleh pinjamnya ini gak?
Apa tuh Bu?
Umi sapa?
Oh iya.
Yo wis ora kenal aku. Karo Umi kan sepedane Bekasi kuwi.
Ya Umi bojo
Kenal Umi akeh iki aku. Akeh ngerti, oh kowe bojone Joni.
yo anak gue itu.
Iya kan.
Awale to.
Si Mbak Ewi kan biyen melu Joni terus ya.
Well,
Iya.
Si Joni ini buka kampung Bekasi ini rembut karo Umi.
Lah iya anak gue Umi anak gue itu awale.
Gua nek gak salah sih menunggu nek gak salah.
Iya, pertama anak buah, sing ngejak Mbak Yune kuwi. Mbak Yune sing dadi pembantu kuwi lho.
Ya, mulane kan ngono.
Si Mini lho, Mbak Mini.
Mulane kan ngono.
Ah, ya tau Joni mah apa.
Kayak ninggalin pas corona.
Kopet, kena kopet.
Kayak waktu kan terakhir kayak cek satu sesuk meninggal ya. Suaranya loading nih kayak acara wisuda juga. Acaranya Andi, aku sing mangkat. Neng jantung.
Aku wis waktu wis 11 kelas kontak.
Acara SMA.
Ya, acara.
Kuwi aku sih ketemu. Saya kan loading. Besok acara. Pas hari-hari itu kayak kata kamu rumah sakit kan. Begini aku kabar Andi meninggal. Lah gue dibayar orang bro aku yang mana? Untungnya dibayar gue si umi kuwi. Beres siapa?
Itu kan pilihannya ya bro. Itu kan pilihan mana lagi?
Punya pilihannya Andi.
Yang anu harapan baru, BP. Mana pilihan dia? Ini tiga lulus terus.
Ya, teko PUP teko apa dulu?
Pindah.
Awal mesti di orang yang udah meninggal mau gimana? Ya terus gimana? Masa utang sampai sekarang?
Ini harus untuk sing keluarganya juga.
Misalnya kayak ngomong doang ini mas, yo kan itu tak kayak apa nanti jo.
Minta aja bukti-bukti apa? Mana ada dulu catatan kita acara?
Kalau ini
sayangnya lah.
Iya, kalau ini. Aku sampai teluas acara loh mas Alper. Makanya aku metu.
Lumayan.
Serah lumayan enggak? Biar mengharian mumpir aku ya. Wis ngedit yo moto yo
Sembarang dengan
nyetak sing ukuran gede yo. Yang kanvasan aku megang juga sing memang garek. Masak lah biar kerja melucu.
nah, Tomcat apa? Kenal?
Kenal, JB ki sing gawe aku ya Mas logone.
Malah. Tapi perasaan aku ora tau petuk sampean Mas.
Aku jarang nek melu Pak Tomson.
Yo, editing itu tak ning kono win.
Maksudnya nang acara wisuda ning
Jarang aku nek acara.
Yen aku sik seneng melu event kan ning Bandung ning Sukabumi ning wis ning ngendi-ngendi.
Yo pisan pindho lah melu kono.
Aku saya kan foto males. Akan bentuk kalau seperti
Kowe kan tak ning Pecenongan ya gawe ngapa Pak Tomson apa aku. Pakai logo JB kan, sih gawe ning panji nih. Ya sarene yo dong ganti ya.
untung. Terus ajine kae coba kae.
Ajine si domu.
Nah.
Yo ora apa yang iso sapa Pecenongan ngerti kan.
Ya sih ini maksudnya juga Thompson ya.
Nek aku melu ngene iki nek Pak Tomson ngerti, aku kadang kayak luwe. Nampaknya beda dhewe. Ya bang melu. Lah, kok ikut lan? Iya, Pak. Nek ini kamu.
Saya yo anu pena omongnya. Sebenarnya
Ya, Pak Tomson ya.
kadang
jenenge rokok orang jaluk. Ngono nek aku langsung meneng. Dik. Satu dik. Rokok yo mesti jaluk lho Mas anggere tiap acara.
Kok yo ora gur sampean to sing ngono.
Yakin. Udah ini tak kick nih sekalian aku lihat HP nganyi. Kowe acara mendino kan ngomong kan dek kan. Acara ini sampai kurang wong mau. Kowe lho tak ngono ya. Acara mendino rokok lho. Jenenge acara mendino kowe rung lah ketemu kowe jurung jaluk rokokku terus. Rung lho kowe rung jaluk rokok.
Yo penting kadang jaluk rokok awake dhewe ra doyan res bae udah. Dek, kucing nek
Umurku yo kowe. Enggak tahu itu ini tapi lulusan ke sepele ke mana deh dibuka. Mesti ngandel nih kontrol itu tuh.
Ya mungkin anu apa modelnya emang pas-pasan menan dibanding di bensin mungkin kan ngono ya. Tapi yo serang disin mah.
Ya yang ngomongnya acara ini terus jadi kan. Kowe acarane ae. Ben awake di jalan bareng ketemu kowe mesti rokok lho mesti jaluk. Langsung menang kopi.
Apa ngedit gini Mas? Aku. Napa edit aku.
Bisa kuat dagang.
Mas.
Ya.
Coba Mas.
Pulau. Jawa.
Jawa kabeh. Pundi Jawane Mas?
Pulau yang sakit.
Sakene pundi?
Sakitnya pulau Lupo.
Lupo.
Iya.
Cara gemolong.
Gemolong ngetan.
Aku Solo.
Tonggol.
Aku mana coba. Solo.
Jadi wilayah Solo lah.
Seragen juga masih sekali sidana Solo.
Iya Solo. Wis kelar Mas.
Hah?
Wis kelar.
Ini final Mas. Ini dulu. Lolos.
Oh, sing dulu kan.
Ketok ya. Dipindahi lantai telu di hall ya. Ketok ya.
Arena telu mau.
Arena telu ning ndi? Nih sebelah. Sebelah ya, sebelah sini.
Di setiap rumah sakit ngirim dokter kok gitu.
Iki campur-campur loh Mas. Ana sing dokter, ana sing peneliti, ana sing dosen, ana sing guru besar, profesor. Aku kene cuma mahasiswa lho, Pak. Lulus S1 ya, gue sekolah IPB. Lawane lawan dokter kok UGM.
Lawane prof ae.
Yang penting kita ngomong. Iya, bener ngomong gini gitu. Bukan menang kita lupa beri kawan Mas. Saya enggak mudik Mas.
Bisa nonton duitnya Mas.
Nonton kok duit.
Terus piye?
Nontonnya niate.
Terus batang aja.
Ya Mas, monggo Mas.
Iya.
Alah, kosek.
Gue bangkune balik rene.
Dijejes di
tapi emang enak. Pantesan gawe apa DPR kan kuwi kok.
Piye kumpulin sih.
Enak kuwi.
Wah, kowe kok ning omah ras do ngedit gur tinggal nggih karo.
Oke, turun iso ngene Mas ya.
Do ngedit enak.
Ora. Kursimu goyang kan wis sempal, sitok.
Terno bulan dia. Kadang-kadang ngomong.
Berarti tadi dalam benar ya, bukan ada uang kamera naik gini, mesti kekir.
Ora. Tadi ini sih, ora akhir-akhir ini yo neng story neng WA ra nongol.
Ya nggak tahu itu kan.
Biasanya Hendrik bulan-bulannya aku aja. Nawar-nawari barang.
Iya bro.
Iya.
Biasanya namanya barang-barang terus ketinggalan ya wayahe update didol.
Nggak nawari
Kanggo
barang.
apa?
50 lah, 100 lah. Tak parani. Serlot.
Iya, yang dipakai dua ini.
Cek satu. Cek satu. Cek satu.
Tadi kan mati hidup waktu ini di sini.
Terus? Ini kena acar ya Pak?
Yang kabel itu.
Jangan alhamdulillah sih. Tadi mati tadi. Sing mati juga tadi.
Yang wirelessnya satu yang sering mati-mati.
Oh
iya, kalau ini memang salah satunya ada yang rada-rada nih.
Jadi suara tadi emang nggak stabil.
Lokasi berarti si Eko berapa nih?